Good morning, everyone. With his blessings, Holiness Jagadguru Sri Shivatreshwar Deshikendra Mahaswamiji. I would like to request Dr. Pujar sir to give welcome address.
Very good morning to all. At the outset, seeking the blessings of His Holiness Jagadguru Sri Shivatreshwar Deshikendra Mahaswamiji. We are all connected online today for virtual seminar and demo on lead optimization of drug candidates using multi-parameter approach, organizing by the Department of Pharmaceutical Chemistry, JSS College of Pharmacy, JSS Academy of Higher Education and Research, Mysuru, in association with Qscr Innovations, Bengaluru. I have great pleasure to welcome our beloved principal, Dr. T.M. Pramod Kumar sir. Sir has been source of encouragement and support in all our departmental activities and developments. So I welcome you, sir, for the webinar. My warm welcome to Dr. Girnath G. Pillai, Scientific Officer, Qscr Innovations, Bengaluru, and the resource person of very webinar. Dr. Pillai has been associated with us on earlier workshop and training program conducted by us. He is an expert in computational chemistry, and we all know he was one of the key resource persons in training CAD software during Drug Discovery Hackathon 2020. Dear Dr. Pillai, we are very grateful to you for accepting our invitation as a speaker and will enrich our participants with the vast experience we have on the topic of the today's webinar.
Thank you so much.
I welcome you, sir. I welcome my dear colleagues, Dr. B. Prashant Kumar, the organizing secretary of the webinar. Also welcome our panelists, Dr. Yogesh, Dr. Dwayanand, Dr. Dikshit, and my other colleagues of the department and college. I am very delighted to welcome the delegates of the today's webinar who have responded very well to our invitation. I am sure that your stay in the webinar till the end will enrich your knowledge on lead optimization of drug candidates by the expert, Dr. Pillai. So this is it about the introduction about all, and welcoming once again everyone. Now I request our beloved principal, Dr. Pramod Kumar sir, for opening remark on the webinar.
Yeah, thank you, Dr. Pujar. Am I audible?
Yes, sir.
Yeah. Good morning, everyone. Seeking the blessings of His Holiness Swamiji. We are commencing this webinar on the lead optimization of drug candidates using multi-parameter approach. The Department of Pharmaceutical Chemistry has been very active, organizing various seminars and conferences, and right now in association with this Qscr Innovation Bangalore, where they have roped in Dr. Girnath G. Pillai, who is the founder of this Qscr Innovations. And just 2 weeks back, on the 3rd of June, the department had organized a webinar on Can Artificial Intelligence Revolutionize Drug Discovery by one of our alumni, who is Dr. Suman Sirmula from University of Texas, El Paso, from School of Pharmacy, USA. He was a speaker there. And again, now we have Dr. Girnath G. Pillai with his vast experience, which he had showcased during the Drug Discovery Hackathon organized jointly by MHRD and Pharmacy Council of India, where a couple of groups from College of Pharmacy, Mysuru and Ooty had also participated, and Ooty won the hackathon as well. I think with his vast experience and exposure in the area of drug discovery, is certainly going to inspire a lot of young minds who would be interested to work in this field. And I'm not getting into the details. Drug discovery is a long process, time-consuming, manpower, and it is something like shooting in the dark. But still, there is a ray of hope at the end of the tunnel. I once again congratulate the department for organizing such type of seminars by the eminent speakers, particularly Dr. Prashant, Dr. Pujar and his entire team. And I wish all the best for the seminar. And kindly excuse me because we have another leadership masterclass which is parallelly running from 10:30. at JSS HER, learning never stops, and we keep ourselves, though it is lockdown and all, everyone is busy more than what we were there during the college hours. So certainly I wish all the best for the participants. There's nearly 114 participants are there. I warmly welcome and take the benefit of this and stay connected, because these are persons who inspire a lot of young minds. It is not just that our college should be benefited. Let the entire society be benefited by such type of speakers. Thank you once again for giving me an opportunity. Thank you. Over to you.
Thank you, sir.
Thank you, sir. Myself, Dr. B.R. Prashant Kumar, secretary and computer for this virtual seminar on lead optimization and drug. I stand before for this important seminar by Dr. Girinath G. Pillai from Just Innovations, Bangalore. So before his talk, let me introduce Dr. Girinath G. Pillai. So Dr. Girinath G. Pillai is a scientific officer at Just Innovations, Bangalore. G. Pillai at So Dr. G. Pillai is one of the coordinators for MHRD's Drug Discovery Hackathon 2020, and is a founder of Just Innovations Bangalore, and is a visiting scientist at Nairo Research India, formerly a Marie Curie Research Fellow at McLeod Limited, Estonia. He carried out his PhD at University of Florida with Professor Qscr and Professor Carlson and University of Tartu. He holds 40 research papers in peer-reviewed international journals, one US patent, and he has co-authored a book chapter on the mosquito repellents. His expertise is in the area of machine learning and molecular modeling, software pipeline development, 9 workflows, and mechanistic driven drug discovery. Apart from scientific positions, he also is a mentor of IIT Kottayam, Government of India's Niti Aayog, ATL schools, BOS member, and a research advisor to a few universities across the country. So such a versatile and dynamic profile, and is one of the young ones who are driving the drug discovery in our country. So I'm very happy to welcome Dr. Girinath Pillai to take over and this seminar come virtual demonstration. My dear participants, remember that in this seminar, there will be an first there will be for a lecture followed by a demonstration, and also Dr. Girinath Pillai is going to issue the license for you to use it for in your personal computers, and you can also benefit out of that like that. So this is the entire program. It is in this virtual seminar. With this, I cordially request Dr. Girinath Pillai to take over. Please, sir.
Thank you so much, sir. Am I audible?
Yes, sir. Yes, sir.
Yes, sir. Yes, sir.
Okay. So I'm going to share my screen. Please confirm
Yes, sir,
if you're able to see. So, I understand that most of the participants won't be able to unmute yourself. So therefore, there might be some interactions going on. So you can basically, what to say? You can actually think of putting in the responses in your chat message. Absolutely fine. I will check the chat messages also. So with all this, I will start. So before moving forward, I wanted to have a small understanding about the audience. So are you all from chemistry or pharmacology, or do you have any other also, just to have a better understanding?
There are blend of students, both chemistry and pharmacology.
Okay.
Yeah, maybe even some teachers also may be there.
Faculties also. Okay.
Yeah,
Great.
Fragment is also.
I understand. Okay, thank you. So I take this opportunity to thank Dr. Prashant Kumar as well as the HOD and the principal of JSS. For the past 6 years, I'm being very well associated to the institute, and every year there is a mandatory visit to the institute during the drug chemistry FDP program. So thank you so much to Prashant sir for considering me for for delivering a session every year, and being missing for the last year, and also we were completely gone to virtual mode.
Yes,
And also we do work closely with few of the faculties in research programs also. One is Sheshadri sir at JSS Mysuru, as well as in JSS Ooty, also being well associated with pharmaceutical chemistry department, as well as Juby madam and Praveen sir. So there's a long relationship, as Pooja sir already mentioned, with JSS. And we do also have students from JSS for internship and project on their MPharm project mainly. So I'm so happy that I'm still associated, and we will have a strong relationship moving forward with research collaborations and others. So before moving forward, please make sure that you address your queries, and it would, if it was, we could interact, it would be much better. But yeah, considering the platform, we also have to see that. Usually, most of the time, what I hear from chemists, the pain points is that, and even I have also faced the same challenges, when we design something on softwares, or when we use some algorithms and we design certain compounds, but at the end of the day, we are not able to synthesize them. So what is the benefit of designing these molecules on computers, and later at the end of the day, you're not able to synthesize them? The second challenge most of the toxicologists or even pharmacologists used to mention is that we try to predict using activity like Qscr or binding affinity using softwares. We were able to predict the activity as well as others. But again, when we go for wet lab experiments like in vitro, in vivo, at least 60% we are not getting as what we expected. 40% we might get. At the end of the day, we at the purpose of moving forward with the research, we end up with some moderate results, and we never got the best out of in vitro. And I'm not generalizing this, for many cases what many people have interacted with me as reported. And what we understood that from the analysis, because being a coordinator for the hackathon, I saw many of the result submissions from across the country. The many things missing was everyone was concentrating only on the numbers. What is the docking score? What is the energy? That's all they concentrated. But we purposefully asked a few questions also. Many did not answer that, and many answered very much not in the right way. Very few answered it correctly. So that is a level of understanding that we have to see what we look forward to in silico based drug design or drug discovery. That has to be changed. Very important. Now, if you think that, sir, we are a beginner level, we might do it after 2 years, after 10 years, I should say that we are already late. When compared to other countries and other research groups, we are already 10 years back. That's what I have to say. When I show the presentation, you'll get to know that. Whatever multi-parameter optimization where I'm pitching now, we talk about it a lot, but we never applied it in our research, mostly not. And this is being applied back in 2010. And why didn't we started applying, or like I know everybody's using Lipinski rule of 5 for all their CAD projects, but why not MPO? That is multi-parameter optimization. So we have to get into that role. And the other second concern, even many of the pioneers in this area, including medicinal chemists as well as in the industry, they used to complain that, not complain, as a concern, as a worry, why we do not have a drug molecule came out from Indian academia. Of course, we do have a lot of nutraceuticals, we have cosmetics, lotions, and all the others. But why not a patented drug molecule that came into the market. Some have gone but failed, but still. So this is something that we have to see. Sorry, I am not aware of. If there is something, please let me know. I'll be happy to update myself. So this is very important and it's very trivial. So ultimately, if, since faculties are also here, I want to say that our publication is mandatory. Being faculty researcher, we wanted to publish and nourish. But at the end of the day, we want to see what can transform into a product, what can translate into a commercializable patent, not simply a patent for our accolades, but rather which can be commercialized. For me, patent means money. If you want to make money, patent it. That's what should be the mantra for you. So keeping all this little bit disclaimers and notes, I will start with my slides. So we will have quite a few slides, but I will try to keep up the time. I'm trying to targeting for 45 minutes. So we will go up to 11 o'clock. So the greatest challenge is how to best incorporate the existing knowledge. That's where what we are. We try to refer a paper and we see what they have done. And then we either replicate the same method and start with a new compound or a new target. Or we try to give some existing knowledge to give a better activity or some enrichment. But why aren't we thinking out of the box? That's where the Nobel laureates came in. Whenever the Nobel laureates express their interest of the particular innovation to their supervisors, they mention it is impossible. But they transform that impossible to possible through research. Because research or scientist means where scientist is basically a person who works on things which are unknown and also want to make things much simple and cost effective. That's the job of a scientist. For that, we have to think out of the box, sometimes out of science or out of the normal science that we have learned or basic science we have learned, but keeping ethics very important. So this is what Martha, who was the head of GSK back in 2009, he quoted this sentence as a big value still now. When you're looking at medicine chemistry folklore, we can see it is a combination of synthesis, structure-activity relationship, receptor interaction, ADME and toxicity. But when you basically talk about organic chemists, they need not worry about receptor interaction or ADME or toxicity. Because when coming to medicinal chemistry, a big role said as lead optimization is into the picture. And that's where the question comes, how do you design compounds? How do you modify compounds based on what criteria? So here, when you're designing or synthesizing a molecule, you have to keep in mind about your mechanism. So when Dr. Prashant was introducing me, he has mentioned very clearly mechanistic driven drug discovery. That is very important. I don't want to call simply drug discovery. Unless you know the mechanism, you talk hundreds and thousands of properties for your drug molecule, doesn't make any sense. So the identification of the right target is always a trivial part. Now, if you ask me, can a chemist do? A little bit difficult because you need to know a good base of the biology, metabolism, metabolic pathways, and receptors. So it is not a one-man army, actually. You need to have multiple people, multiple expertise involved with a good collaboration. So if you think that I can do it myself, I don't think that is a drug discovery project at all. It might be a medicinal chemistry project if a chemist works on. So that's where we have to see multiple people, multiple expertise has to come in together. And when you're looking at, you are solving the biological problems using chemistry-based related expertise, and for medical treatment, and it has to comply with pharmaceutical regulatory bodies also. So a medicinal chemist who is a person who can take the ownership of the compound because he or she is coming out of the idea of optimizations. When you look at the history in 1828 with the synthesis of urea, where the organic medicinal chemistry got started or evolved. But later in 1932, the medicinal chemistry received a formal recognition in academic pharmacy. But when you still look at NCEs, big pharma, I didn't go to the recent data. This history is much more important because when you see every year, the number of new chemical entity or new molecular entity or research molecule that's being approved by US FDA is not that great value or numbers, because it's more challenging. As principal rightly said, it takes many years for us to come out with a drug. But the amount or the research fund they spend on these or any other molecules, even which has not been approved, is still huge. It's about 1 to 1.5 billion US dollars. Do you think everyone gets success? No, some fails also. So when they fail, that is a challenge here, how we can overcome the cost. Now that's where this is a relevant question to all of you. I can see many of the attendees who have already attended my sessions from other sessions also. I'm sure you will be able to answer this question. The question to all the pharmaceutical experts and tomorrow's scientists, basically how you can reduce the cost of a medicine. Rather than telling about government, regulatory bodies, generic medicines, patent and all, tell me as a researcher how you will be able to reduce the cost of the medicine. You can put your responses in the chat because you cannot unmute yourself. I'll wait for 1 minute. Basically, interest of time, we don't want to wait too much of time because we also have to cover many of the topics. So the question here is how you can reduce the cost of the medicine. Hi, Praveen. Good morning. So he said, fail cheap, fail fast. Okay. So any other answers? To be very specific, within your scope of interest, what could be the reason that you can reduce the cost of medicine? As a researcher, okay? I already told you, you cannot answer me with respect to government and other things. As a scientist, as a researcher, how you can reduce the cost of the medicine? You can put it in the chat window. I'm looking at it. But I'm not looking at question and answer, but only on the chat window I'm looking at it. Yeah. So no thoughts? Reduce different stages. Okay, Mahesh replied. Krishna Prasad said the repurposing, including ARCA. Preeti said use of generic drugs. But Preeti, use of generic drugs is always interesting. But a pharma industry require investments to do research. So who will fund them? If you ask government to fund, government cannot only fund pharma. They have to fund many other socio-economic matters. So they cannot only concentrate on pharma. So investors require return of investment, right? So we cannot completely depend upon generic drugs from day one for the research. Reducing the number of steps in the synthesis. Madhura, it could be, yes, that is one way we could. Any other on a pharmacological perspective? So whatever you have said mostly on the synthesis, I agree to it. Ashok Kumar said, share the failures with others so that others don't do the similar mistake. One of the big reason, yes, because when you're publishing, make sure that you're reporting your negative results also. Don't showcase only positives, because a reader wants knowledge from your paper, and discussion part is the brain of the paper. So make sure that you not only report the positives, you report also the negative. So others also know, okay, I shouldn't waste time on that because already someone has already worked on it and failed. So make sure that at least in the supporting information, you report all the failed compounds, or even synthesis also, whatever failed, please mention it there. It's a great value that you can share with the scientific community. Reduce the times of failure by accurate procedures. I mean, so Chirag is telling about the strategies. Okay, do virtual evaluation before synthesizing. Subhankar mentioned this, but Subhankar, keep in mind, all virtual evaluations are not correct. If you do the virtual evaluation in the right way, it is correct. So make sure that we do it in the right strategy. Shyam Sundar said, develop new technologies to discover new medicines with less time and less expenditure. Okay, thank you so much for all your answers. So let us move forward. Everybody has come into some conclusion about somewhere related to what I'm going to discuss. As rightly mentioned by one of the participants, cause of failure. If you can reduce the failure, that's where you can reduce the cost of medicine. If you are not reducing the failure, the cost of medicines will go up. And this is completely dependent upon the researcher or the scientist. Don't depend upon technology, and others. Let's imagine that whatever the FDA approved drugs before 1980s, okay, after 1980s only most of the tools, algorithms came in. Before itself, we had challenges. Early 1980s, most of the design of molecules came completely from human intelligence and brilliance. Nothing on software, nothing on in silico evaluations. So even based on that, they were able to come up with many drugs in the market. But still, the failures were mainly because of pharmacokinetics, ADME properties, and efficacy. So you can say 40% was on ADME and 28% was on efficacy. If you want to read more data about this, please go through the two review which I have cited in this slide. That's where you will learn about before 1980s, what were the failure reasons for the drug molecules approved or rejected by FDA, especially oral drugs. But when you go post 1980s, that is 2000 to 2010, again, we have two different reviews from Nature's review on drug discovery. So here we could see things have changed because post 1980s, we had implementation of statistics. We have Qscr, Qscr, and many more models, validated models, experimental studies. You could see that many papers on ADME wet lab studies, experimental data came later than 1980, just because they found that failures was mostly because of pharmacokinetic properties. But then we had sophisticated tools, models, and then we were able to reduce the challenge on ADME properties as well as efficacy. But then we started bombarding and exploiting with all these tools, but we forgot about strategy. We really don't know, are we doing it in the right flow or not? The biggest disadvantage here is we just follow tutorials. We just follow a paper. What they have done, we just simply replicate it. We never thought of doing it in a different way. I'll give you an example. When I was doing my PhD, most of our Qscr, what we do, click a button, get all the descriptors, correlate it, and then try to say, this is my Qscr model. I did with 10, 15 modules, and I couldn't get any of the very meaningful Qscr model. Not simply R square is Qscr. You need to have a very meaningful model. But then after a lot of discussion, failures, what I did was I did a DFT calculation, confirmation analysis, and nowhere it is written there, confirmation analysis. Manually, I had to calculate the distance between carbon and nitrogen where it could form cyclization. And based upon the distance of that carbon and nitrogen for a pre-organized conformation, I consider that distance as a descriptor. And then I generated a model. And that model was having a better accuracy than any other descriptors I chosen, which is already standard available. So if something is not really working out, we have to think ourselves and come out with something innovative. And that was published back in 2013 on Chemistry European Journal. I need not to say how good that journal is. So we were able to predict that model, and many people are now using that model to predict the relative abundance of cyclization. So it was nothing related to biology, but totally into chemical reaction. So the strategy is very important. So don't think that whatever came in with tutorial or like a single paper can be replicated for your work. It depends. It might be, it should be optimized. And optimization is unless you try, fail, again try, you cannot understand where to be optimized. So you need to have a lot of time and patience for that. And if you say, I don't have time, then you don't have time to do research also. That's what you have to take it. The next challenge was toxicity, because we were mostly concentrating on affinity. We were mostly concentrating on how to improve affinity, how to reduce the binding energy, increase the concentration to picomolar or nanomolar. But we never thought about toxicity. Even if we think about toxicity, we mostly think about cytotoxicity or maybe a few others. But there are many other things that should be taken care of. Cardiotoxicity, carcinogenicity, and many more. So toxicity is also one of the biggest failure on oral drugs post 2000. Now that's where how to solve all these or how to materialize all these things to understand a bit early, like Praveen said, fail fast, fail cheap. So if you can know all these things much earlier, you will fail. You will fail in the sense you fail early, so that you don't invest too much time and money on that at the later stage. So that's where we want to know the role of statistics and machine learning. So we have a software called a StarDrop, where deep learning and machine learning is involved in structure-activity relationship. One of the biggest challenge in drug discovery is that we have sparse data. We don't have very good data. Even the data is good, we still have experimental errors. I used to tell most of the time during my presentations that even in our pre-degree or even our +2, we used to learn titrations and we repeat our experiments 3 times, right? Because our human error, eye error could be there. So we repeated the experiments so that we can find out the deviation. But when we grew up, we were scientists. Due to the fund limitations or other restrictions, we were not able to repeat our experiments. Rather, we carry out only once, and then there will be experimental variations there. So these things should be identified because there could be false negatives also. So we need to have a strategy to identify these experimental outliers, and also screen multiple endpoints to target the best compound for synthesis. I don't know how many of you have noticed that. Same compound, same activity, let's say anti-malarial activity. Different groups will be reporting different endpoints. Now the question is which one I should consider as the valid one. So we can use machine learning and deep learning algorithms to come up with finally which is the best endpoint for the compound for synthesis, based on analysis of several other factors on optimization. So when you use machine learning, you can always look at the disease prediction, structure analysis, drug repurposing, and novel drug development. But when coming to novel drug development, let me be honest here, AI is not very much influenced in drug discovery. So don't worry if you do not know AI much. But ML has its own good position in drug discovery. So learn first statistics a lot. Statistics is the bread and butter for machine learning, not Python coding. So don't worry about that. I had another session where I was telling about person who does not know coding can still do machine learning using KNIME and Orange software. So we are hosting, and the KNIME company, German company, is itself hosting drug discovery and a life science session on July 7th. And I'll be moderating that session for India, for the country India. So there we'll be discussing more about KNIME, how you can do machine learning using KNIME for drug discovery. Now let's look at the evolution of CAD. As I already told you, early 80s, it was classical metchem. We didn't have any tools, but later it got evolved. But when you look at the drug discovery phases, the initial phase is called target identification. So when the interns work with me, especially the chemists, they found it very difficult, this particular part, because they were not learned or trained for biological systems or understanding mechanism or metabolism. So they had to undergo a lot of reading and maybe not find the right data. And based on questions and discussion, they ultimately end up with the right one. So we wanted to identify which is a catalytic function or which is the right key amino acids that you can interact to upregulate or downregulate a specific mechanism. Unless you disturb or enhance the mechanism, your drug discovery has no value in many of the diseases. I don't say every, but many. Then we look at the lead discovery and lead optimization with many of the techniques that we are already aware of. And finally, we go to drug development based upon preclinical trial test where we have ADME, toxicity, and PBPK simulations. And finally, we go with clinical trials. Fortunately, all these methods could be assisted with computer-aided drug design. But let me warn you here, do not consider computer-aided drug design as a replacement for in vitro studies or in vivo studies. Never, ever consider that. Computer aided, the word aided means it's not replacement, it is assisting. So the computer-based assistance to make decisions. It never said that to replace your wet lab. Now that's a challenge that you have when you want to publish your paper in a very good journal, Very good journal, I meant means not high impact factor, high value paper. So I know a journal called Chemical Biology and Drug Design, I think, CBDD. It is having only a impact factor of 2.2 or 2.5. But in order to get a paper accepted there, it's a tough time. It's not that easy for your paper to get accepted in that journal. It's from Willie, right? So it's not about impact factor, the value of that journal, and how much your paper gets cited by others, not by you, by others. That evaluates how much is your research value to the community. Now, if you ask me what is the success stories of CAD on pharma, there are more than 112 compounds which has gone to clinical trials, phase 3, phase 4, based upon computer-aided drug design approaches. A very beautiful blog which talks about basically the fragments and all. So fragment-based drug discovery is also looked at. Can you hear me? Sorry, in between something happened. Am I audible?
Yes, sir, audible. There was slight
okay. Great. Okay. Yeah, I thought that's why I asked you. Great. Thank you so much. So this particular website will tell you about the success and failures of fragment-based drug discovery also. I'll come to that point a bit later. Now let's look into drug design. This is where it's very, very trivial. Based on the availability of the data, you have to choose the method. It's not that I want to do this method, let me do something on my project. No. When you have a 3D structure of the protein from X-ray crystal, that's where you have to do molecular docking. Now you will say, sir, I have homology model structure. Fine, but make sure the homology model structure, your catalytic site or the functional site or the binding site has a good coverage and identity with your template. If that region does not have any similarity with your template, then your output of docking result is not reliable, because homology modeling itself is error prone. And on top of that error, you're adding another docking prediction, which will be a several magnitude of error. So make sure that what you're doing, see, it is not that others did and they published and let me also do. No, first try to find out whether you're doing is right or wrong. I have seen papers in Nature where they have used homology modeling. And I could see that last week that paper is retracted now, which published 8 years back. So that's how it is. So make sure that we do the right things always. So it is important that based upon the availability of the data, you choose the right method. And next, ligands. Coming to ligands, mostly it is recommended to use X-ray crystal structures of ligands. But we know that most of the compounds might not be crystallizable, so they don't have in the position to CST. But then what we do is we take the 2D structures, optimize using OpenBabel or anyone or RDKit, and then we try to generate it the 3D structures, and then we do docking and generate the conformations. Absolutely fine. The problem here is we all know that when the compound is as such on a natural, like on a normal stable situation, it has a particular geometry or a conformation. And let's imagine that is safe to consume. After consuming it, bind to biological systems or maybe macromolecules, the conformation will change. And that conformation is called bioactive conformation. So the process of doing molecular docking is not to find docking score and binding energy. The process of doing molecular docking is to find the optimal bioactive conformation of your ligand, because that conformation will change. And we want to find an optimal active conformation, bioactive conformation, because that should form a stable complex or a co-crystal with the protein. That is our aim for doing molecular docking. It's not just simply score. So in hackathon also, when we were looking at that, everyone concentrated on the explanation on the scores, but very few talked about the catalytic site. Very few talked about the conformations of whether it could be stable or not. And ultimately, after doing molecular docking, it is mandatory that you have to do molecular dynamics. Now, if you say that I don't have computers to do molecular dynamics, don't worry. I'll be sharing links at the end of this session where you can do molecular dynamics for free on the cloud using Google Colab to a certain extent with some limitations, but still you can do. But if you say that, no, I don't have any softwares, it is expensive. Let me tell you, all molecular dynamics softwares are absolutely free for academics. Number one, Gromax, Amber Tools from Amber, then NAMD, the only software which works in Windows, that is NAMD. Others all are in Linux. Then you have Desmond. Desmond, if you go to DShaw, you will get the academic version of Desmond with the Maestro, which is again absolutely free for academics where you can do molecular dynamics. And finally, Charmum Command Line. All these 5 softwares which is available across the globe, which is for biomolecules or macromolecules, they're all free. So there's no investment on software rather than you need investments on your hardware. This is what we call it as structure-based drug design. And I said molecular dynamics is very much mandatory because docking does not give you accurate results. Let it be any docking software. Because molecular docking dynamics finally tells you on a dynamic situation how far your drug can stay stable in the complex when you make your protein flexible. And keep in mind, molecular docking is not an irreversible. It's a reversible process. So your drug should go and bind the protein and it should get released. So that should be a reversible reaction. That is also important, which we can understand from molecular dynamics. So that is the trivial part. And when you think that you have the 3D structure of protein, and when you don't have ligands, means you tried docking with all possible ligands, nothing worked. That's where you do fragment-based drug design. But please keep in mind, I always tell this, try to have an organic or a medicinal chemist sitting next to you when you're doing de novo scaffold hopping. So if a biologist is doing de novo scaffold hopping, I said no to it, because software can come up with any fragments based upon synthetic accessibility and feasibility. Don't imagine that 90%, even 90% it is synthesizable. I can give an assurance of 80% based upon some data that I received from commercial pharma industry. Up to 80% it is synthesizable, not more than that. So make sure that a chemist sits with you when you are doing fragment-based drug design, which is very much important. And when you think that you don't have the 3D structure of the protein, even homology modeling, and you have only the ligands, then try to do pharmacophore, similarity studies, and Qscr. Working a lot on Qscr, let me tell you, one of the dangerous and unknowingly making mistakes is the only method in computed drug discovery is Qscr. You can make a lot of mistakes, even you don't know that you're making the mistake. So be careful while you're using Qscr. One simple thing I'll tell you, when you're doing 3D QSCR, everyone texts, I want to do only 3D QSCR, but there is a big challenge here. What is that 3D? I clearly told you, when you're doing QSCR, you're talking about a biological activity or bioactivity. When does a compound becomes bioactive? When it achieves a bioactive conformation. Are you calculating the descriptors for that conformation? Or are you calculating a descriptor simply for a 2D to 3D converting software? From that 3D structure, you are calculating the descriptors. I made that mistake earlier, back in 2010, but then I corrected it. So initially, I also made that mistake. So we learn from our mistakes, right? So then like Professor John Dearden, search online, a very famous professor in Liverpool University. He pointed out my paper saying that this is wrong. He wrote a critical paper on that. And then we wrote a rebuttal. And then myself worked with the Professor Dearden on another paper. Together, we shared an authorship. So that was a healthy criticism, which helped me to learn a lot from him. So that's how it should work. So make sure when you're doing 3D QSCR, either you use protein docked conformation, which is confirmed that it is a bioactive conformation, and use that conformation to generate the descriptors. Then if you think that there is no proteins available, try to do a pharmacophore conformation search and then generate those 3D structures to calculate the descriptors. So that's why I always recommend everybody to do 2D QSCR. There's no bias about that, anything. 2D Qsr is the best and without any bias situation. That's where we call it as ligand-based drug design. And when you go to combinatorial drug design, this is there in high throughput screening. If you want to read a little bit more about this picture, you can check my PhD thesis. So this is from my own PhD thesis I have taken. Now, when we talk about molecular docking, still we use the terminology, it is a key lock hypothesis, which was pinned by Emil Fischer from 1894. But let me tell you, this is only half truth now in 2021. Multiple keys can fit into a single lock, one by one, when you do docking. And multiple locks can have a fit from single key. And that's where we call it as drug repurposing, right? Now, I think you can make it make sense now. Usually, key lock hypothesis, one key can fit into only one lock, other than the traveling locks, which we use. That is a different purpose. So the key lock hypothesis is only half truth now because things have changed, perspectives have changed. So we also have to change the way we look at molecular docking. This is a picture that I've taken from Enzyme Methodology book. And it has a very big value on what you can achieve from molecular docking. I never asked here what is its score, but I asked few questions like, as a normal human being could ask for molecular docking. Do you know where the drug will go and bind? Do you know how the drug will bind? Can you tell me how strong the drug could bind? And why the drug is binding? Or without binding, why can't it be there? What makes a drug binds to the receptor better than the others? Similar these questions. Once you answer these questions itself, your discussion part of the docking will be ready. But otherwise, most of the papers, what you have seen is they write about interactions, this amino acid, that amino acid, this is the bond distance, this is the energy, and this is what is the atom it is interacting, or score or energy. But we have many more things to discuss. For example, catalytic function. That is where the mechanism comes into the picture. We wanted to know what is the role of the key amino acids in the protein which correlates to that function. That is catalytic function, or it could be motif also. Later, if you're targeting an enzyme, we wanted to understand whether you're targeting an active site or allosteric site. This question was also asked during the drug discovery hackathon. Many or very few answered it, many just left it. So they don't know the mechanism actually, but they just did the docking and reported it. So we need to know the mechanism actually. The third one is cleft detection. Again, another big thing that gives some mistakes. By using the software, you will be able to predict the pocket. 1, 2, 3 pockets will be there. The first one is always the largest pocket because having a larger volume. Now, how many of us check that whether that particular pocket contains all the key amino acids in the catalytic function or whatever we are going to target? Because unless you're targeting that amino acid, there is no point of talking about that is my right cleft. But vice versa, what we are writing in paper is that my compound is having interaction with these amino acids. But what is the role of the amino acid? Very fewly I have seen mentioned. So that's where we have to recreate what we are doing with molecular docking. Don't worry, because this won't be mentioned in any of the tutorial, because tutorial is not a research document. It is a step-by-step protocol document for a software. What I'm talking now, how you apply that into a research. So make sure that whatever pocket you are identifying, you have the key amino acids there. And then geometrical analysis. You want to understand, is there any conformational changes when a ligand goes and binds there? Then flexibility. Are you going and targeting the substrate bound site or an apo site? Very important when you're doing anti-cancer studies and on conservation studies. Selectivity, specificity, and sensitivity. How confident you are that your compound will go and bind only to that specific binding site, not to any other binding site of any other proteins in our biological system. In fact, you're not going and docking with all the proteins. You're docking only with 1 or 2 proteins and reporting the data. So if a reviewer is raising this question, how do you answer? You don't have an answer because you didn't analyze it. So it is very important to understand how you can find similar binding pockets of the protein that you have interacted in order to find with others. So in Qscr, we have, Qscr is a software which I'm going to demonstrate. There you have a function called as, you can search similar binding pockets across the PDB from RCSB website. And that's where you have an answer for conservation studies. Surface characteristics. Are you targeting a lipophilic or a solvent accessible area or a negatively or positively charged regions? And at the end, that's where we talk about interaction energy based upon solvation and desolvation terms. So this is something that is very important that you have to take care when you're doing docking. Now, let me tell you, I'm been using this slide for the past 6 years. Hope Prashandas still remember, because this was the same slide I've been using when I used to visit JSS for my presentation. I still never changed. But even after giving a lot of presentations and talk, I don't see people are using this to transform, put into the research. So make sure that if this is really good, or if this really makes sense, start implementing in your research. Initially, it will be hard, but later you will find it very easy. That's where you will see the acceptance rate of your paper will shoot up like anything. I am against paid publications, because there are many beautiful journals where they don't take any money from authors like RSC, Valley, Elsevier, even ACS also. From the readers, of course, they take money. But again, many of them have access to it. Otherwise, many other routes to get access to the paper. So those things are there. So try to high aim for better journals and publish on that. And when you're looking at docking, we also have to understand about tautomerism, wrong element assignments of preparation of ligand or proteins, and pseudoproteations. And also addition of hydrogen is also very important. Most of the time when you click a button, it just adds hydrogen. That's what is protein preparation, right? But how many of you know that the orientation of the hydrogen is also important? If the orientation is different, your interactions are also making different. In your pocket, the orientation of the hydrogens of the amino acid, if it is not the right way, that is going to be bad. So these things are to be taken care. So clicking up one click and then software does everything means, then why we are sitting there, right? So we have to make sure that this does the job in the right way. And what does happen when you do docking? When you have all these green sticks are amino acid, and the white sticks are the complete compound, and the red one is the fragment. So when you place a compound into the pocket, it tries to fragment the compound and start different conformations, and then try to generate all possible poses to identify the best bioactive conformation. And that's where we wanted to quantify with the affinity and binding energy with the delta G, where the compound is unbound in water, and delta G when our compound is bound with water. So basically we are calculating delta G to understand the binding energy and binding affinity. But in order to understand this, we want to completely look at the hydrogen bonds and desolvation terms. Now, this is something similar to MMGBSA, where in MMGBSA you require MD trajectories, MD simulation trajectories, but here no parameters or limitations. So it's more of a statistical. Now, if you ask me accuracy, molecular dynamics itself is not accurate. So the same way you have that accuracy compromise here too. So why no dock score? Because I did not discuss about dock score at all. Keep in mind, docking score is only used for virtual screening. I'll clarify that now. When you have, let me see if, I'll clarify in this particular slide and I'll go back. So when you have 45 lakh compounds, you want to do a virtual screening. How do you rank the best top 1% or 2% of the compounds? Only based on score you can do that. So scoring tells you how comfortably your compound is fitting into the pocket. That's all. Nothing biochemical, nothing interaction, nothing strength, nothing. Nothing is considered. Just looking at how the compound is comfortable there. So for a 45 lakh compounds, the best way to rank is just to click the button, sort it, and choose the best top 1%, 10%. That's it. For that, docking score makes much more sense. But after that stage, when you wanted to do further analysis to confirm among these 2,200 molecules, how many of them are having interactions with the key amino acids which I wanted to target, number one. Number two, what are the energy or affinity parameters for all these 2,200 compounds? Number three, whether whatever the conformations that generated within the pocket, are they crystallizable or not, which is talks about torsions. And four, the lipophilicity. Until unless these four parameters are being satisfied, we cannot then narrow down our pool of compounds or hit molecules. So finally, we ended up with 974 molecules. From 974 molecules, available compounds, which is purchasable, is only 172. Now, when I talk about 974 compounds, there will be docking score for that. I don't say, but don't expect that docking score will be in the same order what we got for dock score. So for these 172 molecules, the topmost structure might not be having the best dock score. So don't think that always the best dock score is your best compound for your synthesis or for testing. That's why you ended up with false positives. So there are, apart from score, these are the four parameters that you have to consider for that in energy terms, not in score terms. But in some software, the score is also called as energy. So that you have to check their manual to check how it is being addressed. Now let's look at how we can quantify that which is crystallizable or not, or which could form co-crystal or not. These are the dihedrals or the torsions. When it is in green color, the green color represents that it is present in Cambridge Structural Database. Cambridge Structural Database is a crystal structure database for small molecules. Similarly, if it is yellow, that means it's unusual. Red means very rare. And then for a medicinal chemist, what is the advantage? You want to have some information to do lead optimization. So if you get an information saying that this particular carbon has a negative contribution, means it is red color, it is bad for the affinity. That's where a chemist will think about, okay, let me try to optimize this or make some changes which is synthesizable, and then redo the docking and see if it is improving or not. So in docking itself, a medicinal chemist can apply their optimization techniques. And finally, enrichment factor. I don't know how many of you know how to calculate enrichment factor, and this is the equation for that. So enrichment factor is mainly for virtual screening. If you're not reporting the enrichment factor for virtual screening, the quality of virtual screening cannot be accessed. And I don't know how many of you have done this, because in drug discovery hackathon also, we asked this, many of them did not report the value. So that is what we are missing. We are doing a lot of things, but we are not reporting the right thing, what the community requires to reproduce your data. Until unless one knows the quality of your virtual screening, nobody is going to take your data for further research. So this is something that we have to seriously look into it. Enrichment factors and all way back since 2007-6, it's already there. But are we using it? Are we reporting it? That is a question that we have to ask ourselves. And these are some examples on the compound which is interacting with different two portions of the pocket and to understand the different energy functions. And this is something called, as I told you, the fragment-based approach. If someone is working on PROTACs, okay, PROTAC is not new, old, but something emerging in everyone's mind to talk about the linkers between two different ligands. So you will be able to find out the possible fragments. So we do have a fragment database from Zinc and PDB, and as well as CCSD. And then based upon the pharmacophore constraints, we will be able to identify the possible fragments and then merge them to form a possible compound. In order to choose which will be the possible fragments or which type of chemotypes to be chosen, that's why I recommend a chemist is always recommended. And that's where the compound gets formed. Gleevec is the first anti-cancer drug for breast cancer. And then very important slide moving forward when we look at discovery, drug discovery, because so far we were looking at finding out a hit molecule. That's it. A job for bioassay, potency studies, molecular docking is just only to find a hit molecule, not a lead molecule. Once you find a hit molecule, next is what you wanted to do, the lead molecule. So when each of the expertise work independently, They have their own limitations and advantages. Computational chemist mostly focuses on predictions. Chemists will mostly focus on better yield, shortest steps, and better purity. But when coming, there is a mismatch between them because they don't agree with each other. Modelers will say that I designed a very good compound, very good activity, no toxicity, blah, blah, blah. But then when it is being proposed to a chemist, chemist will say, no, it is very difficult to synthesize. I cannot synthesize this, very expensive. Reagents, I cannot get it on time. The chemist will synthesize whatever they can synthesize, and they'll give it to a modeler saying that, can you do something and give me some docking result? Now, where is the target? What is the target? What was your intention to design this compound? So the strategy itself is a problem here. A medicinal chemist should have a problem statement in front of her or him that I'm going to target this particular target, and from that target, I have to design my molecule. But in earlier days, right now, many of the things have changed. So in earlier days, they already decided to synthesize a compound. Later, they tried to see which target it could fit in. So there is always a miscommunication and gap between these two expertise. So that's where we have to see how we can look at the importance of speed with productivity. When you go for proposal itself, make sure that the chemist as well as the modeler works together and double check with the virtual results, and then finally successfully go with the synthesis. That's why Thomas Kruse Lipinski rightly said, let the medicinal chemist or organic chemist takes the co-ownership of the idea, because they are who designing and making the compounds on table. And that's where the co-ownership of the idea. So we have to go in hand. It's very important. So just using software, don't predict anything and claim that I found something. No, never, ever. So we need a better situation where the chemist and the computational people could work hand in hand so that they each other understand. I'll give you some examples in the coming slides. So that's where Lipinski said a rational dialogue between a chemist and a modeler can be very, very productive. So don't think a biologist can design a drug and come out with a drug. No, you need a chemist there ultimately. One of the important slides for throughout my slide is this one. So I already told you we've identified heat molecule from a set of chemical molecules which having a good potency. Now, don't tell me heat molecule is one molecule or two molecule. There should be at least a few hundreds of molecules to call it as heat molecule. Unfortunately, we never go for bioassay studies for 10 or 20 or 30 molecules because of the funding, because of the time. So I think you have to take a pledge now. Tomorrow, when you're writing to funding agency, tell them that you want to test 60 compounds. They will not give money for 60 compounds, but at least they'll give money for 10 compounds to test. That should be your strategy. When you wanted to confirm heat molecule to lead molecule, make sure at least testing 10 to 30 molecules and with the diversity. Otherwise, you end up with moderate activity, medium activity. This is what we see in many papers, right? Because they couldn't achieve what they wanted to achieve as per the aims and objective of the paper. So we have to look at an optimal balance of all these properties. When it transformed to heat molecule, we also wanted to call it as a lead molecule when it satisfies toxicity, human industrial absorption rate, metabolic stability, solubility, flexibility of all these compounds. Now, when I say metabolic stability means not predicting a Ki value of 2D6 or 2C9 of isoforms of cytochrome P450. What I'm trying to say here, we wanted to understand whether your compound will be stable unless it reaches the target of that particular drug. When it passes through liver metabolism, if your compound breaks down, it will not reach the target and have the binding as you saw in your docking analysis. So it's very important that your compound should be metabolically stable. By doing bioassay, you're not doing that, right? You assume that it has gone to the target. To that, I'll come a bit later, but that's a big bias there. So you have to quickly identify the situation when such balance is not possible. So fail fast, fail cheap is the mantra. Only when confident, move forward with the compound and avoid missed opportunities, which is very important again. So unless you avoid missed op means, for example, we use Lipinski rule of 5 everywhere, but where to use, I will discuss in a moment. Now, this is something that helps a chemist to understand which region of the compound is prone to be toxic or which is non-toxic without affecting the activity of the molecule. So this gives information to the chemist that, okay, this is the region you can think of optimizing. For example, A and B compound. A is more toxic. A is very active. B is moderately active but non-toxic. Now, ultimately, a medicinal chemist will choose only the compound A for optimization because A is very active, because the activity is a most priority top property for drug discovery. So I have to take A and then I have to optimize A because in order to reduce the toxicity. If I give it to a chemist, medicinal chemist, and asking, can you please reduce the toxicity? Can you do some optimization? The chemist might ask, okay, where should I make modifications? When I make modifications, activity could be compromised. How can I maintain the activity and only optimize the toxicity? And that's where you have these knowledge-based and machine learning-based predictions, which tells you the prediction based upon earlier failures and earlier success rate. So it's not that simply simulated based predictions. So now it's a question time. Let's imagine that you have 10 different molecules with you. I know some of the attendees already know this answer. So let's imagine that you have 10 different compounds with you, and you have to carry out 5 different experimental assays. In order to choose the best 2 molecules from this 10 compounds, right? What is the methodology you have to implement to choose the best 2 molecules? I'll repeat it in a different way. You have 5 different experiments to be carried out for 10 different compounds. 5 experiments have to be considered because that is what makes a decision what is a drug molecule or not. I mean, drug-like molecule or not. So among the 10 molecules, I want to choose only the best 2 molecules for the next set of analysis. How to choose this best 2 molecules considering all the results of 5 different experiments? That is my question. Most of the time what we do experiment by experiment. So for one experiment, among the 10 compounds, it is easy for you to choose the best 2 molecules or best 1 molecule. But when I'm saying you have to consider all 5 experiments in order to choose the best 2 molecules, I don't know whether we have tried this earlier or not, because I'm heading to the technique called as multi-parameter optimization. So in MPharm, in pharmacology, as per PCI syllabus, in your drug discovery paper, you have a terminology called multi-parameter optimization. And that's the only way that you can solve this challenge, right? So let me go through a few slides and then we'll soon go through the demo. So we have a lot of data for the same project. We have a lot of data from in silico, in vitro, and in vivo. And when we are moving forward to prioritizing it, the prioritization is depending upon importance of which experiment is more important, which experiment is less important. Means all experiments to be considered, but which is more important and less important. And second, priority based upon uncertainty. For example, you have an experiment, but the results are not much reliable. That means very uncertain. Don't ever consider that result as a top priority. That's why I told you, when you do homology modeling, we really know that there's a lot of error in it. And then you're using that as a main structure for doing docking studies. Your results will be still unreliable. The same way in multi-parameter optimization also. Any property that is unreliable or the results are having more error, don't ever give high importance, because that is an error which is going to reflect in your result also. And finally, you look at the selection. When you look at the selection, we have a bad habit. Clicking the sorting, ascending, descending, the top 2, we select, this is the best one. Yeah. But coming to chemist as a lead optimization or making modifications in your compounds, quality is not the only factor that you have to consider. You have to consider diversity also. You need to have heterogeneous structures. You need to have different chemotypes to make modifications. If you try to choose only the same derivative, same analogs, you end up with failures only sometimes. If that particular analog is successful, all will be successful. But if that particular analog is failing, all your derivatives are going to fail similarly. So it's better that you have diversity. I don't say that you don't include everything, but at least have different chemotypes at the initial phase of screening. And this is the best example also to tell you that how we go with the prioritization techniques. The prioritization techniques, we look at the potency. Let's say we selected few compounds and we did potency test. Few compounds are removed based, they are inactive. And we selected some compounds which are good. Later, we did absorption rate studies and metabolic stability studies. On each step, few of the compounds are being removed because they are not good based upon a threshold, expecting that you're getting a very good compound. Unfortunately, they are not very good compound. You end up with a bad compound. The reason is your good compound might have a good potency, but the absorption rate might be bad. So it is being thrown out. So the best way is that you have to be very cautious when you're doing experiment by experiment filtration. You might miss opportunities. This is what I told you earlier. Avoid missing opportunities. To all this, the only solution is multi-parameter optimization. Multi-parameter optimization is used back in 2010 from a PNAS paper and for CNS drugs. And it's being proven very successful, this methodology is. But are we using it? Yeah. If anyone is using, please let me know. Back at least 2 years back, because since 2 years, I know many of our customers started using, but I don't know about others. So when you look at the rule, this is where I want to talk about Lipinski rule of 5. Everybody uses the first filtering criteria for downloading database, and we use Lipinski rule of 5 to filter the compounds to choose the best molecules, and then only we do docking. Some, they do docking, and after that, they will do Lipinski rule of 5. The question here is, Lipinski rule of 5 tells you that molecular weight should be less than 500. Now, do you think a molecular weight of a compound with 501 molecular weight is a bad compound? I don't think so. You're going to say that 501 molecular weight is a bad compound, because FDA approved drugs itself, we have something there up to 850 molecular weight. So how can you avoid a compound with 501 or even 510 molecular weight? So I don't say Lipinski rule of 5 is bad, but don't use it in the way that it has not to be used. Means you took a database, first do a virtual screening using docking. You consider Lipinski rule at the end. That too only for oral bioavailability. If you're working on something on CNS, again, has to be reconsidered. So make sure how you're implementing certain filters always. There are many FDA approved drugs which does not comply with the Lipinski rule of 5. Right? So we have to make sure that how we use this. And that's where multi-parameter optimization comes. So in Lipinski rule of 5, how we are going to use in multi-parameter optimization. I'll give you an example. Any compound which is having 0 to 500 molecular weight, I will give them a score of 1. So in multi-parameter optimization, the scoring is between 0 to 1. That's it. Nothing more than that. So for anything less than 500, I'll give 1. Then I will say any molecular weight between 501 and 600, I'll give a score of 0.7. And any molecular weight between 600 to 700, I'll give a score of 0.5. And anything above 600, I'll give a score as 0.2. That's it. What it tries to tell us is, when you simply apply Lipinski rule of 5, any compounds greater than 500 molecular weight will never be considered in our database. Nothing. It is done. They are totally removed from our subset. But when I give the importance level, like score up to 500, 1, up to 650, 500 to 600 is 0.7, 600 to 700 like 0.5, and anything above 700 will be 0.2. There I'm giving importance. So anything less than 500, I have the most important factor there. Those compounds to be given more important. But other compounds from 500 to 600, I'm not removing them, but I'm still giving a lesser importance compared to the compounds which is having less than 500 molecular weight. The advantage here is that till the end, we keep this compound still in our data set, subset for further analysis, because sometimes it might come up later. Because just because molecular weight is bad, you don't say that the compound is bad. So we need to have a strategy to see how this compound is well balanced in other properties also. It's not only about molecular weight a compound is said to be bad. I just gave an example. Like that, many other properties are there, right? So this is how we apply multi-parameter optimization. And this is the paper I was referring to for the CNS drugs. They applied MPO and they found it successful because they were able to find 14% extra compounds from the Visa candidates by using MPO. That 14% extra means they were the missed opportunities when they were doing the traditional method of filtering. And this paper was published in 2010. And now we are talking about 2021. Are we using it? I don't know. That's why we are looking at SAR approaches. A combination of all these ADME properties all comes from experimental, theoretical, and empirical. Due to the interest of time, I'll quickly go through. So in Qscr, all these things you might be knowing, but very important factor. If a paper comes to me for reviewing for a Qscr paper, I'm being reviewer to ACS, Willie and all. If you do not mention, I just reject it. That means you do not know what is Qscr. Because plus or minus error is very important when you're writing an equation for Qscr. If it is a regression-based model, but if it is a classification-based model, then we have different other validation techniques that to be taken care, or ML-based model. So it's very important you talk about error. Have a practice of talking about standard deviation until unless there's none of your predictions are reliable. That's what I used to tell. In calculator, 1 + 1 is always 2. But in science, we have 3 different answers. 1 + 1 is equal to 1.9 plus or minus 0.1, 2.0 plus or minus 0.1, 2.1 plus or minus 0.1. All 3 answers are completely acceptable in science, but not in mathematics, right? So that's a way that we have to look at these predictions and statistical methods. The only thing that I want to focus here is that I got many questions from students and others. So I took the same compound, tried predicting ADME properties using paid software, free software, online portals, and all gave a different value of solubility. Which one should I take? It's a very relevant question. The only answer to them was, I didn't never claim that paid softwares are always the best prediction, never ever. Or free ones are the best always, no. It all depends upon the class of compounds used. So let's imagine that this is the training set of the compounds, and that is the applicability domain. If my compound, which is colored in yellow color star, is able to give a prediction within the applicability domain of the training set, and the standard deviation is very less, I rely on that prediction. And if another compound is very much close to the applicability domain of the training set, it's kind of an okay situation. Still reliable, but you have to be careful. But if the standard deviation of another compound is far away from the training set, or the standard deviation will be much larger, that tells you that the class of compound or the chemical structure of this compound is way different from that of the training model. That is unreliable. That doesn't mean that the model is bad. That tells you that the model does not contain the similar structures what you have submitted for prediction. And the software which can able to give standard deviation and which talks about the applicability of domain of applicability, that is much more reliable, because that clearly tells us whether our compound class of structure is there in the training set or not. Until unless your class of structure is in the training set, a model cannot predict your the activity for your structure accurately, at least accurately. So that is what we have to look at. The scientific validity of a Qscr is defined like endpoint, applicability domain, mechanistic interpretation, and physical significance of descriptors to activity. Now, it is not a question that I used Qscr modeling with 3,000 descriptors, 8,000 descriptors, and all. No. The question here is, finally, whatever descriptors you got, do you know the meaning of those descriptors? Do you have a proper physical meaning that can be correlated to your activity? Otherwise, your Qscr doesn't have any meaning. So make sure that it's not only about R-square and error, it's also about how to relate your descriptors meaning with the activity. And that's where you have to keep the phrase called causation is not equal to correlation. You can correlate with anything, but you need to know the cause. You need to know the physical significance, how this is being correlated. So Keeping in mind the ADME properties, now you might think how the 2D structure of the chemical compound can predict the ADME, because there are certain physical chemical properties which has a physical significance to the ADME properties. That's how we predict it. So these are the ADME properties available in StarDrop, like aqueous solubility, partition coefficient, 2C9 isoform of P450, Hurg liability, blood-brain barrier, human intestinal absorption, plasma glycoprotein, plasma protein binding category, and few other thermodynamic parameters. Then we do have some models for predictive toxicology from US EPA. And then direct nexus is something called as human toxicity endpoints. There are 43 human toxicity endpoints which is approved by FDA. So here again, it tells you this region is carcinogenic. So it tells a chemist that, okay, you can think of optimizing this region so that we can avoid carcinogenicity of this compound. So these are the 43 toxicity endpoints, human toxicity endpoints, not animal, human toxicity endpoints based upon clinical trials from the pharma consortium. So we do have mutagenicity, genotoxicity, mutagenicity, then mitochondrial disruption, cardiotoxicity, organ toxicity, and many others, including neurotoxicity and respiratory. Then we have something called as P450 module, which talks about regioselectivity, which tells you which atom of the molecule is prone to breakdown or which could form metabolite at different isoforms. So P450 cytochrome has different isoforms like 3A4, 2D6, 2C9, 2C19, 2C8, 1A2, and 2E1. So the algorithm will be able to predict which your compound is prone to which particular isoform and which atom is prone to breakdown. So this is that particular nitrogen, we said it's breakdown, the carbon is forming a keto group here. So it tells you the liability of the compound, which that's where we take a pro-drug strategy. And for this chemist, especially for medicinal chemist, we have something called as medicinal transformations, which will be able to predict the new derivatives or transformations based upon the property requirement. For example, you know that this particular region is toxic, so you can define that this region to be replaced, and it will be able to tell you which are the predictions having lesser toxicity. Now let me quickly give you a case study, and this is what we are going to do a demo today. So the first case study is that you have set of molecules with this biological activity data. We wanted to do a R-based group or activity neighborhood analysis. What it tries to tell you is, with a change in the functional group from, let's say, fluorine to chlorine, it will analyze and tell you whether your activity increased or decreased. From a side chain of carboxylic acid to methoxy, it will be able to tell you whether your solubility increased or decreased. So that kind of analysis could be done here. So that is called hit triaging. So based on an existing data set, you can do this hit triaging, and that's where you optimize the new molecule or the existing molecule with you, because you know that if I change this functional group, there could be a chance of increase in activity. But again, it is a chance, right? So, and also we could link between the docking studies as well as the activity studies. And bioisostere replacements could be done. Pro-drug strategy, because when we know that that nitrogen carbon region is prone to breakdown, so we can come up with a pro-drug strategy. Then transformation, where we think that we have lactam in our system, but this lactam might be prone to some property issues. So we wanted to transform it to pyrimidine. So when you transform it to pyrimidine, the question here, most of the chemists, okay, I don't have SciFinder. How do I find lactam to pyrimidine synthesis route? These transformations are already published. So we have only the published data available with us, so that you can refer to these journals, papers from JMedChem or heterocyclic chemistry to see how the transformation from lactam to pyrimidine is being carried out. Similarly, we can do optimization to reduce the cardiotoxicity, that is Hurg, where this particular carbon is replaced with carboxylic acid. And you could see that the PIC50 of Hurg reduced from 6.7 to 5.2. And if you do not have SciFinder with you or Reaxys with you, we have something called as ICSynth, which also uses online interactive support for retrosynthesis pathway. So whatever design molecule you can give, it will be able to predict the literature data as well as a similarity on the yield to tell you what could be the possible retrosynthesis for this compound. The same way the SciFinder does. Don't expect that exact compound references will be there because your compound is novel. So don't expect that exact will be there, but they will be telling you the similar compounds, which is already reported. And similarly, you have something called, this is something new approved by NIH for their research, which is called explore a pharmacophore of a hit molecule and we match the pharmacophore of new compounds. I'm not going to explain too much, but I want to just tell you about the output from Merck. So Merck took this compound and took this software, and then they were analyzing it on 17 billion chemical space compound space. And they were applied to 12 different drug discovery projects. And they said that 80% of the compounds that is predicted by these chemical space was synthesizable. That's a very good accuracy that they can achieve on a prediction base. And IPY design is also protected. That means whatever the compound they predicted new, they're all intellectual property, so they are able to patent them. So this is all about these were some of the papers that is published by considering all the factors which I told you right now. Yeah, most of them are from my own group, and this was a collaborative work, as well as these two are collaborative work, but these two from our group itself. So my current focus is on aging and longevity, and we are in a discussion with the AI department of JSS for collaborative work with the health department for AI and ML projects. By that, I would like to conclude, but if you have any questions or any other things that we can take now or even after the demo. So it's up to you. Now, I will be posting some of the links which I have shown here in the chat window. So let me, yes, already Dr. Yogesh already posted that. Thank you so much, sir. So what I want to tell here is a few videos and others are there. So whatever today I presented, all the slides are available in the slideshare.net/GiriBio. It's there in the chat window. The links are given. And then if you want to go into some of the demos, let's say students might not have money to buy softwares, right? So you need to get access to freely available tools. So for docking, protein modeling, and many of the few of the ADME and other conformer analysis using 9, I have put it in my YouTube. And then if you wanted to work on, as I told you earlier, if you do not have good computers to perform molecular dynamics, I have already uploaded Google Colab. Google Colab is something free for everyone. You can run GPU-based molecular dynamics calculations. And the Google Colab is already there in the workflow pipelines link, which I have given. Go to MD Notebook and go Colab. So that there I have done it using GROMAX. You don't need anything from your mobile, also you can run it, but you have to define the PDB and all those things are there. A little bit of understanding of Linux is very important because otherwise you cannot customize it. And only challenge there is every 12 hours, the system gets reset. So any calculation happening within 12 hours, it's fine. Otherwise, you have to pay. And that will be like per day, you can do 154 nanoseconds per day can be done on Google Colab. And if you're interested to know the success stories of CAD in FDA drugs, you can go to the podcast, which already Dr. Yogesh has already put that link. So these are some of my free time hobbies and activities which I'm trying to do. So if you have any questions related to the presentation, please shoot it up. Otherwise, I'm directly going into the demo.
Okay, dear participants, thank you, sir. Please, if you have any question, you can put it in Q&A box so that sir can clarify.
Okay, they don't have any questions. That means you did not understand what I told you then. I did not explain it properly then. No, I'm just kidding. So please feel free if you have any comments or disagreements or agreements, anything is fine. Or shall I proceed with the demo and later you will ask the questions? How do you want me to? So you can type on the chat and you can let me know so that I'll continue.
I think we will just wait for a few seconds, then later.
Yeah, sure. Okay.
Fine, sir. If they have any questions, maybe in between they will ask so that we can clarify at the end.
Fine. So there is a query from Krishna Prasad. Which tool would you suggest to understand ADME characteristics of accurately for phytoconstituents? Krishna Prasad, I already told you, you use any tool. Which tool can tell you the standard deviation of ADME prediction? I prefer that only. So personally, even if it is free or paid, make sure that, so otherwise, let me tell you, I'm telling you that PKCSM is the best. Okay, and you predicted it, and ultimately you did wet lab studies. You don't see anything is coming in par with the prediction. So tomorrow you'll blame me. Giri said this is the PKCSM is the best tool, but I couldn't get anything good. That's exactly what I'm trying to tell you. Every software contains different chemotypes of the structures used in the training set. And if I'm claiming that I've used all the structures in this world in my model, please, you shouldn't believe that. I might tell, but you shouldn't believe that. Because there are new structures are evolving. How can I include everything into my model? Never ever possible. So make sure that whenever you are getting a better like a standard deviation, which tells you how far your structure is within the chemical structures used for the training set of the model, that gives you more validity or justification. What is a good pKa value for hit molecules? Dr. Basavaraj, that's where many of them, when they ask me a question, which is the best score? What is a range of binding energy? It all depends upon you within the data set. Are you going to consider 17 billion compounds, which is so far available in this world for your docking studies? Then I can answer this question, which should be the good pKa. But when you are selecting only thousands of molecules, few of them, you might not have explored all the pKa space, only a specific one. So just based on pKa, you shouldn't make a choice. It completely depending upon many other factors also. So within your safe space, which one is the best? That's what you have to relatively look into it. Okay, which software you suggest for confirmation analysis? Arun Kumar, for confirmation analysis, there's a free tool called as Balloon, and then you have something called as RDKit. But the challenge in both the tools is they use molecular mechanics for confirmation analysis. So just with molecular mechanics, the accuracy is very bad. So make sure that after generating this tools, you should make sure that we are also doing DFT based calculations. So that is very important to confirm which conformer is more stable. Another question, Naga, see, going through these links as a learner or as a beginner is not enough. You have to read a lot of reviews. As a beginner, don't ever read any research publication work. Please read only reviews. In the reviews, they mention about what is good, what is bad. With a case study, they tell you with this case study, this will be better and that will be not better. So for a beginner, always review is the best. Let me know the basic book to understand Qscr concept. Eunice Pasha, there is a lot of old books, even by Andrew Leach and few others. So Qscr, I don't recommend to learn by book because many things have evolved now. New techniques have come. I don't know recently any new authors wrote about the recent techniques. Otherwise, we have Indian author, Professor Kunal Roy. So you can also look, he more focuses on the statistical part, not on machine learning, but machine learning parts are already available in Medium blogs and all. Yeah. How to predict the active site of the homology model protein? Venkatesh, again, a homology model protein active site, two ways to consider. One, you have to identify the key amino acid using ProSite. ProSite is an online tool. Another thing is that using pocket finders or binding pocket predictors, and see that whether the amino acids in the binding pocket predictor, as well as what is being said by the functional site or motif or catalytic site predictor database, all these matches each other, including if there is any literature data available. That's where you confirm that is your proper binding site, which includes the key amino acids also. Arun Kumar, for DFT calculation, we do provide turbo mode, but for free, you have something called Orca, but it works only on Linux. Qscr, no, I have never used it, so I have to look into it. So I have never used Qscr. So another question, as a beginner, do I understand this sub? Okay, what as a beginner I have to do to understand this as a subject better? So I already told you this, you have to go through your reviews, read as much as review on that particular topic. And after that, in that review, whatever research papers they have referred, and after reading the review, go to that paper and understand the case study, then only read any other research work, so that you will have a better understanding about the concept. Is there any software which suggests a suitable binding receptor for the known chemical structure? Absolutely. It is called HitPick server. So Divya madam, there is something online server called HitPick server and SCS server. These two servers, if you give the SMILES of a compound, and then it will try to find out the suitable binding receptors, including binding DB. So I have written a code like a workflow in 9, where you give the the structure as STF or SMILES. It will search binding DB to find out what are the similar structures and its PDB that is being reported. So that is available already. Great. So I think I answered all the questions. So let me go to the demo session now. So if you have any questions, please stop me. I'll check in between. I won't be able to keep my chat window along with the window what I'm using now. So if there is anything in between coming up, please let me know. I will answer them. Okay. So first we will go through StarDrop. So StarDrop is more of a ligand-based drug design tool. You can load SDF file, SMILES, and CSV files with SMILES. Automatically, the structures will be drawn by the algorithm itself. The software, we do have perpetual licenses, but for the training, for all the attendees, when Prashant sir shares the details, we will be able to share the licenses with you so that you can at least try what we are trying to learn today. Now, don't try to compare this with existing other players. This is entirely different and cannot be compared, because the features that is here is not there, and what the features they have, it's not here. So this is something completely for lead optimization, pharmacology, and especially for hit to lead identification and optimization step. Now I just loaded only these 3 columns. Number 1, structure. Number 2, the ID column, which is the name, and then the predicted biological bioactive data. These are the 3 data which I had in my SDF file, and there is around 124 compounds, as you can see in the bottom row 124. So I had around 124 compounds here. Now, what when I just simply clicked on this, automatically the statistics of that particular activity came in. I could, if I mouse over it, I will be able to see there are 27 compounds between 4 and 6 bioactivity, and there are around, if I come here, I can see there are around 92 compounds between 6 and 8, and if anything above 8, I have around 2 compounds. So if I select something here, automatically it is interactive. It will automatically select the compounds in the table also. So the graphs or graphical representation is completely interactive. Now there is something called as chemical space. Okay, don't confuse between clustering and chemical space. Chemical space is something that I'm defining completely for all this structure based upon structure similarity as well as representing based upon different properties. It's a combination. So chemical space can talk a lot more information than a clustering analysis. Anyway, so before moving forward or coming back to explain about this, just a second. Okay, so before explaining this, I want to tell you different models available. As I told you, there are ADME models available within StarDrop. So this is aqueous solubility, then 2C9, blood-brain barrier, plasma glycoprotein, and plasma protein binding category. And if you mouse over it, it will tell you all much more details about it. So it is if it is saying low, that means less than 90% of the compound bound to plasma protein. And if I mouse over to human industrial absorption, it tells you if it is plus means it indicates absorption is greater than or equal to 30%. If it is minus means it indicates absorption less than 30% for a compound. So it talks about two class categories for the human industrial absorption. And if I mouse over it again on HERG, it will tell you it is a Heather human ether go related gene potassium channel inhibition. Now, very interesting thing here is that if you go to the help menu on the user guide, it's common like any other software. They talk about how to click here, click there, what is options I know. But here we have around a 200-page reference guide, which talks about science. Basically, earlier I told you, we need to have a better understanding about the type of compounds or chemotypes of the compounds that is being used for the prediction of models and what is accuracy levels and all. So those kinds of informations are given here. So let me go to that page directly. This is only talks about the science related to it, nothing related to options or anything. A lot of reference details and others. So let me go to that data. Yes, ADME model references. So this gives you from which experimental data all these properties are generated. Now you might think, sir, this is, if you give all the information, then we can also do it. No problem, because until unless you know the quality of the model that we are using inbuilt, you cannot rely on any data. If someone is saying this is secret, then I cannot believe it, because we want to rely on the predictive capacity of the models. That is very important. That is one of the things that you have to disclose in Qscr also. So when you're publishing papers, let me quickly show you that. When you're publishing papers, this is the GitHub which I was telling you. Let me go to that aging data. So when you're publishing papers, make sure that not only the like supporting information, make sure that you're also giving all the data, raw data, to the scientific community. So if you download this external prediction Excel sheet, download it, and it will be asking you a certain descriptor value of your compound. Just enter the descriptor value on the respective cells. It will predict the anti-melanogenic activity automatically. So we didn't build a website, but we make it very simple offline. So this is what we have to do. When you're doing a model, it's not for publication alone. Someone else has to use it and predict using a model. So if you just download this external prediction Excel sheet, and then all the other data are given, structural file, statistics, all statistical details are given, applicability domain details of the models given, which descriptors we have used and what are the descriptor values, all is being disclosed. Because sometimes we cannot upload these many files into the literature. So what we did is we gave the link in the paper and we said you can use these models for your structures for the prediction of anti-melanogenic activity. So that is the purpose of Qscr models. So until unless you disclose all your model details, who will believe that this model has a capability to predict the values? So this is something very, very important whenever you are doing a Qscr modeling analysis. So here you can see that like around for log p value, around 2,950 compounds were being used for the training set with R square of 0.92. So the accuracy level here it is 0.92 for predicting the logarithm of octanol water partition coefficient for a neutral compound. Now, as I told you, we have also wanted to know what kind of compounds being used for this prediction. So we were able to use neutral compounds, acidic, monobasic, polybasic, phenolic, and citrionic. And this database will be improving year by year. That's where the improvement of the model gets increased. And here you can see for a different property, acid, base, and citrions. And what are the acid functionalities are also given in Smirt's filter. So this gives you immense information when you're writing an explanation for your ADME properties. Nowadays, what we see in ADME property prediction, one graph and a table, which tells you these are the ADME properties. But what analysis did we do? Did we do any kind of statistical or machine learning based analysis? No. Most of the time, we just give a graph saying that a radar graph, which is again available in the free tools, which tells you that, okay, this all properties fits into this radar. But is that enough? That's what we have to explore and see. Okay, so apart from these models, we do have something custom models like Cacao models for skin permeability. Then we have detention time, Kovacs filter for order, fragments and all. If you're working on those models are also available. And if you think tomorrow that you want to do some modeling with this tool, of course, we can, when you publish it, we can put it in the repository so other people can use it and cite your models also. That facility is also available within StarDraw. Right now, these are the tox models which is available, which I told you the US EPA developmental toxicity models are also available here. Right. And then you have other human toxicity models, and you have the pKa predictors are also available here. Most acidic, basic, and all pKa is also available here. Now, when you click this button, it will be able to predict all the values. So here you can see log S at like aqueous solubility at pH 7.4, log P, and others. Now, these are the values here. Now, where's the standard deviation? So you can click this particular statistics button so that you will be able to see the statistics. So here you can see for some compound it is showing 0.621 as standard deviation. In some compound is 0.70. In some compound for log P it is 0.43. So in most it is 0.43 because it's very close to the applicability domain. And some it shows infinity, means that compound is not in their particular vicinity. That means within the applicability domain. So this is what the information tells you about uncertainty. Until unless we have these compounds in the particular database, then otherwise it will be difficult, or the models will be difficult. So most of the structures that I've used here is already available, right? Now, if you think that tomorrow I'm going to invent a new or novel molecule, of course, it won't be there in any software that you're currently using, but the similar substructures might be similar in some of them. So that is what we have to look into. So that's where one point. The next point is about the multi-parameter optimization, which we were discussing a lot, and that is a topic of interest for our today's session also. So here the pKi estimate, which is again from bioactivity, I said it should be greater than 7. Let us do an exploration here. So when I double click on it, I can see, as I told you earlier, in the case of molecular weight, anything less than 500, I'll give a score of 1. Anything between 500 to 600, I'll give 0.75. 600 to 700, I'll give 0.5. And then anything greater than 700, 0.2. So like that, I can give the scores here so that it will be considering a few others. So I can click insert, and I will say minimum value 7 and maximum value maybe, minimum value as 6, maximum value 7, and I am giving a score of 0.5. And this, correct. So what here I have done is any values between -infinity to 6, I'm giving a score of 0.05. I don't want those compounds, but still will consider in my data set because I don't want to miss them. Anything between 6 to 7, I'm giving a score of 0.50. Right? Anything between 6 to 7, I'm giving 0.5 as my score. Anything above 7, that is something that is acceptable for me as 1. Now, as Dr. Bhavinder, like last time asked, which is the right range for pKa? Now, you might also ask me the same question here. How do we know which is the range that I have to choose? How do I know what is the topmost value or least value? This is something that you have to, two ways you can take, either from existing literature for your set of activity or biological activity that you're looking at, because there they will be giving the range. So when we publish papers, we also mention these could be the optimal range. Next, or in the same case, you can look at trial and error based upon the data set space that you have. Otherwise, there is a multi-parameter explorer algorithm, which is a patented technology, which will be able to identify what could be the optimal range within the data set you have. For example, you have compounds with 50,000. Within that 50,000, which will be the best range, it will be automatically able to predict. It will not tell you that, okay, by standard, the Ki value should be this one. No, nothing like that. Within the data set, that is important. So that is an algorithmic based. Otherwise, based upon literature or experience data, we will be able to give these values. So does anybody have any questions so far? Yes, someone is having some questions. 24 out of 15 was active. Okay, 24 out of 15, confusing. And 2 active molecules. I went to time skill study against MRC. Okay. One was taken lesser time and the other took more time, but both are different derivative. ADME prediction showed some toxicity. So what would be the next step? But I have not done the bioassay against a targeted enzyme. Also for beginner, how to understand Qscr better and any paper for reference. So Niladri, Qscr paper to understand, I recommend reviews. Okay, I have to promote myself, right? So on 2012, 13, I have written a review on Qscr with my colleague. So that is one thing that you can start. In 2017 or 18, I think, there is a very beautiful review, around 32 pages, which talks about why, how, what Qscr. And it is by the main key players of Qscr in the world. Paula Grammatica, John Dearden, then Alexander Tropshaw, all these people were there. It's a very beautiful written review. That's fair enough for you to understand which is the best or how better you have to start with Qscr to understand. So these two reviews is something my very personal, but otherwise many others are there. But don't miss reading these two reviews because we have clearly pointed out certain things. Now, when looking at your work, you already mentioned that you have not done any bioassay. So that is something important that you have to do. I know you have looked at a time kill study, but activity data study is also very important. Number one. Number two, even the ADME showed a toxicity, but did you do any cytotoxic studies or any other toxicity studies? It would be great. I don't know whether extensive molecular studies to be carried out in this case or not, but check that out so that your paper, and if these compounds are synthesized, I think so that you have made these compounds, I mean, synthesized these compounds, and if they are novel, and if it is crystallizable, do the following. Number one, try, if it is crystallizable, crystallize at least 1 or 2, submit in CSD database, it's free, okay, before publications. So submit it and mention that in your publication. Publish in at least JMedChem, aim for that. If they reject or do not accept with comments, go for the next level. So, but aim for JMedChem, and then you go for the others. And third, if your compound is very novel, even after molecular studies, cardiotoxicity, cytotoxicity studies, including activity studies, if it is really good, make sure that you can talk to a pharma industry or advisor to ask this only one question. What are your expectations in order to get a compound converted from academic research to an industrial research? What all data you require? According to their requirement, try to get that data. If you have fund and if you have time, and then patent it, including all those data, patent it. Then we can see what happens. So these are my recommendation. Again, clause, disclaimer based on funding and based upon the time. So otherwise it would be good. That's what Niladri, I said, it's all depending upon what you want to do. See, PhD is a training exercise. It's not that you have to do everything at that spot itself. It's a training and learning time is PhD. So for your PhD, this is fair enough. But if you think that you want to do something out of the box and then complete it with much better reach, that's what I suggest. Completely depend upon your fund, time, and supervisor's interest also, because until he or she does not have funds, it will be difficult, right? So it depends upon that. So, but no need to be so stringent that you have to do that. Maybe probably for your PhD, you have already ready with whatever work you have done. So it depends upon that. So I cannot strictly say this has to be done. It all depends upon many supporting factors. Anyway, so since you don't have any questions related to the software, I'm moving forward. So this way you will be able to put the scores and other things, right? So now I will cancel it so that I will use the standard one which I was using, where I said any value up to 7, I'm going to use 0.05, because I'm sure that if the activity is not good, how can I use this compound for my further analysis? And then anything above 7, I accept it as 1. So this is what I have done. Now, if you think that I wanted to add a different criteria for my analysis, let's say I want to include, let me check, pains count. So I can click and drag and put it here. And then I will say anything less than 0.5 is acceptable for me, because pains count should be 0 actually. So that's what we look at. So anything less than 0.5 is acceptable. That's what I'm going to give, and not very important, because there's a lot of conflict for the pains count property. So that can be also given. If you want to delete this, click this and delete. Now, let's say you optimized, a person who is doing optimization did all this optimization. You can save this to a file and you can share it with your colleagues for them to use the same multi-parameter optimization. By default, we do have some of the multi-parameter optimization profiles available by standard, like even molecular Lipin's rule of 5 also. But for this project, we are using this one because we considered activity data, solubility data, industrial absorption, partition coefficient, cardiotoxicity, 2D6 and 2C9, P450 data, plasma glycoprotein, plasma protein binding category, and blood-brain barrier, because we are using a non-CNS drug to understand how far this is not having blood-brain barrier. And then we were able to calculate by clicking this arrow, we will be able to calculate it. When we calculated, we got this particular graph. So here the score, as I told you earlier, it is between 0 and 1. Here, I want to show you something. Yeah. So this is where we wanted to do the analysis. Let me sort this compound based upon the list of compounds based upon the highest bioactive compound. This one is the highest bioactive compound, that is having 8.142. Okay. So just looking at the activity data, you will say this is the best compound because this is having a highest bioactive data. Now, when coming to the scoring, we have 0.29, but let's try to again sort this based upon the highest value. Now you can see something different. Your compound, the compound which we expected that bioactivity is good, which is 8.142, is having a score of 0.29 only. And it is now in the 20th rank. But when I go up, I can see another compound which is 7.52, having its bioactive, is one of the best compounds while considering ADME property as well as activity data. Now let's do an analysis. Why did the compound which is mostly bioactive did not come to the top list? First, when we are looking to this compound and looking at the ADME properties, we could see the log S here is 4.18. In fact, hope you remember, in the scoring, we have given the high affinity is of more important. The next highest important is about solubility, which is having 90% of importance. So when I come here, this particular compound is having 7.52 because my criteria was the compounds which is having greater than 7 bioactivity can be considered. So here it is 7.52, still good. But the solubility here is 4.185. Let us see to the compound which we had selected that this is the bioactive, which is 8.142. What happened to that and what is the solubility for that compound? So when I click on that, you could see the solubility here is 3.83, even though the bioactivity is more than 7 and the highest, which is 8.147. Now you might think just based on solubility, you did not consider this compound. No, there are other reasons also. Erg liability, the cardiotoxicity. For this particular compound, which is highly bioactive, is having 5.6, right? Now if you go up, for this particular compound, the best one, it is less, 4.55. Now you might ask me, so you told me that these are all predictions. There are errors, there are standard deviation. So how reliable these results to make decisions? That's a catch here. Make sure that all these analyses are not for choosing the best molecule. All these analysis intention. So any computational drug designing approach for selection of molecules, don't end up selecting 1 or 2 molecules from computational studies. Make sure you have at least 10-15 compounds for each computational study filtering. Per se, we are trying to say that we want to remove bad molecules from our system. I'll explain this using this chart called as snake plot. I'm very confident to remove all these compounds from my data set, all these compounds. The reason is the score is too less, like less than 0.2. The reason is if my score is too less, that means many of the ADME properties are not satisfying. Because I want optimal balance of all the properties, then only I can consider that. So when you look at the color here, any of this red color tells you that they are having better activity data. Anything the color fades and comes to yellow, the activity data is less. So whatever the compounds that is coming on the top, they do have optimal activity data as well as other ADME property. That doesn't mean that it is non-toxic. It might be toxic also. But it is a group of compounds that we are selecting in order to the next step of lead optimization. But we do not consider these set of compounds and waste our time. So the process here is to remove the bad molecules from our data set rather than choosing the best molecule from our data set. That shouldn't be your intention when you're doing molecular or computational tools. That's exactly what it replicates to docking also. When you're doing docking, if you end up choosing 1 or 2 molecules for your wet lab studies, you end up with failures and it won't be really great. So make sure that you choose at least 10-15 compounds for your testing. If you don't have funding, ask for 30 compounds or 60 compounds for funding, and let them give you for 10 compounds. Absolutely fine. But when you write only 2 molecules for testing, okay, 2 molecules. So this is what should be your intention when you're doing these kinds of analysis. That's why in my title, I mentioned drug candidates, because these are all drug candidates. But as my stages proceeds, I want to reduce my space, means number of compounds to be reduced. I can only tell the best molecule when whatever molecule enters into clinical trial, those are my best molecules. And those molecules did not come from just with the computational predictions, but with many other wet lab validations. So up to then, any questions? Shyam Sundar, while comparing small molecules with peptide therapeutics, which one is more significant? Shyam, when you're going to peptide, yeah, it's a completely a different science when compared with small molecules. So peptide is good, I would say, and many experts used to say, based upon natural peptides and synthetic peptides, that's where we have to see. It will be more toxicity and other parameters when compared to small molecules. Yeah, so that's what I want to comment on peptides. But when you bring peptides into many of these tools, any of these tools, including other players, they consider peptide as a chemical molecule because on the ligand side, peptide are still considered when a force field or like any molecular dynamic simulations being applied, docking and all. But for ADME properties, there are protein descriptors available even for peptide, but we don't have models straightforward with an accuracy to predict it. There is one online server available for ADME properties for peptides, but accuracy is to be questioned. Anyway, so now let us come to our main task. When you go to design and when you found that your most active compound is having some issues with ADME properties, now our task is to how to optimize them. So I will first try to optimize with our this particular hERG liability. Okay, what we will try to do. If, for example, I think that I can make the compounds. So what I will do is I'll make this carboxylic acid. Okay, I made it as a carboxylic acid. Now let's see if there is any change in solubility and hERG. Yes, we can see. When I converted it to carboxylic acid, I could see the hERG reduced from 5.6 to 4.0. And it affected the solubility. I think it was 3.6, now it is 3.4. No, it increased actually. It was 3.0, I think, earlier, and now it is 3.4. So this is something that a person should know. This is synthesizable, then only you can do it. But let's imagine another way. Let me reset it. Yeah. So let us imagine another way. I do not know chemistry. So if I know chemistry, I know where it could be synthesized, I'll be trying to design and see what could be the prediction. But I do not know chemistry. So I wanted to see what software will be able to predict us for reducing the hERG liability. For that, we will be going to NOVA is the particular algorithm. Then we click this arrow button. So here we have 4 different options. One is chemistry transformations. Chemistry transformation is that a specific region of the molecule only can be transformed into another with a specific synthetic routes. And then we have something called match series analysis, molecular match pair analysis. That's what in some cases it is called. Reaction-based library enumeration. So you can define the reaction reactants, and then you will be able to generate all possible products. That is reliable too. And then we have scaffold-based library enumeration. Again, it's a fragment-based approach. So there you can define the fragments, and then a virtual library can be combined from fragment-based. So now we will do chemistry transformations. Now we have to define which region of the compound has to be replaced. So what I will, and you have to mask from any transformation. So I'm trying to mask saying this particular region in order to tell that this region should be untouched and no replacements has to be made. Since I already know that that region is having key interactions and that could be affecting the affinity of the bioacid. So whatever the highlighted region that they will be trying to replace. Next. Now it will, let me click on show applicable. So there is a BioStir reactive database, which will be telling you what are the possible transformations available for this kind of choice of bonds that we have given. So So based on the choice of the bonds, it will be able to choose out which are the possible transformations. So we have only this much possible transformations available. Otherwise, the list is a bit larger. Now, if I go to this one, I can see here isopropylamine to piperidine, right? Now, if I click to manage, it will be giving me all more information about the literature data. So isopropylamine to piperidine were reported into all these papers, and it is already said that it is neuroleptic and dopamine receptor antagonist. Or if you're going with some other combinations, let's say for this one, and then here you will be seeing piperidine to tetradecahydroacridine, and here it is already reported as analgesic, and these are the following literature references. So this is the way that you can do chemical transformations. Okay. By the way, I'll select all of them to see how better we are getting the predictions. Now, coming here, it's very important. Two things. Remove compounds with undesirable structural features, and you can select the features. Means these are, if you mouse over it, you will see these are all taken from literature data. So let me show you that. Yeah. So you can see this is taken from this particular literature, which tells you this is one of the biggest disadvantages of fragment-based drug discovery, because it will come up with all possible regions, and later we come to know this is toxin, this is mutagenic, this is actually genotoxic and all. So there are already databases available saying that these regions or these fragments shouldn't be in the drug molecule. So we can consider and select those molecules or fragments so that this will not come into your predictions. And then I want to select subset of each generation. So I am trying to say, I want compounds
Hello, Giri sir. Okay. Okay. I think,
so can you hear me now?
Yes, we can hear.
Oh, great.
There was some interruption to the internet.
Yes, the internet got disconnected in between.
Yeah.
Sorry. Okay, so I gave the name Herg, which we are creating a name. So I'll go back. So I was trying to say that we selected what all the fragments that cannot come to be part of the replacement, and we are selecting the each generation wherever the compounds having lower Herg value. That is my ultimate goal. I'm selecting only 100 compounds. I can increase the generation, but it takes more time. Due to the interest of time, I'm not giving that more. It will take more time for calculation. So I'll give next. Here I'm just giving a name Herg and then finish it. So it will take some time. It just started. It will take some time. Yeah, it's already there. That's why I said it will be much faster if I give only generation 1. Now, let us look into the table form of this particular structure. Yes. Here I can see my Herg original for the parent compound is given here, 5.622. And here I'm going through each of the compound that is predicted by the software, which is having a Herg of 4.69 based on the criteria that I have given. It would have predicted others also. I didn't give any value. I just said lower Herg liability. So it will predict all possible ones. So here, what is this? Let us look into it. Here, the software automatically itself trying to tell us to transform from isopropylamine to piperidine. And these are reported in all these literature work. And another one, what they have said is tertiary amine to cyclic phosphonate. And they have also given literature references. So this way, you will be able to again take these compounds to docking. Now, these compounds are non-existing. Please keep that in mind. Even we can search online also. So these are called transformations, which is synthesizable. And the success rate here is about 90%. I'm not talking about bio-isosteer replacement. I'm not talking about that. I'm talking medicinal transformations for improving the ADME properties. So do you have any questions at this point? Because I'll be showing a few more details also. If we are masking one area of the molecule and modify the remaining part, will it not affect the binding of the molecule to the drug binding pocket? Absolutely. Whoever asked this question, it's absolutely right. That's what I was explaining when I'm telling you, you have to know which. So when I was masking itself, I told you, because I am aware that region is having some interaction. So I'm not going to select that. I'm going to select the other one. Now, if I'm making the transformation also, of course, it is understood that there will be difference in activity. So activity will be changing. I cannot guarantee that it will be maintaining the activity there itself. No, it will be changing for sure. So that's where it is a iterative process. So I don't know whether I showed you that slide. Let me just pick that up. Just a second. Yes. This particular slide talks about that only. I didn't go through that because time was up. So I took around the 3,100 compounds virtual library, did an in silico ADME profiling, again, based upon docking and analysis. And then I did a multi-parameter optimization on a CNS penetration. And I selected only 10 compounds based upon score and diversity. I gave importance to score 25% and diversity about 75% importance. So for example, in 10% means it's around maybe 310 molecules, right? So if I'm giving 310, in that 25% is based upon the quality of the score. But 75% I've given diversity. It means I want different class of compounds to be that 10% what I'm going to choose. So this is something that we also have to look at. Sorry, it is 31 compounds. And then measure in vitro potency. Then I did wet lab studies on 31 compounds. And then I score again to balance ADME as well as potency. So even though I know the activity of this compound, but still I redid the activity based upon the diversity. So I calculated the score again, including ADME and potency data. Finally, I chose 25 compounds from 31 to score and diversities. So, but here at this point, since potency is also involved, I gave importance of 75% to score and 25% to diversity. But again, I'm going to iterate this once again as soon as I optimize it. So optimization step is an iterative process, and you try to find out which one is more optimal with balance of activity as well as ADME. And that is the reason why drug discovery takes years to come up with the best optimal structures, only just because of this iterative process. Right? I hope I answered that question. Okay. Another one is like, for anti-cancer compounds, what are the important ADME properties to be concerned analysis in publications? Shyam Sundar, it's nothing like only for anti-cancer or something. Of course, when you're going for virus, antiviral drug or infections on skin, again, skin permeability, and also when you're looking at CNS drugs like blood-brain barrier. So those very, what to say, topical drugs, no, we have to look at specific ADME properties. When you're coming to anti-cancer drugs, it's mostly what we are looking at. It shouldn't kill our normal cells. That is what we usually do with MTT assays. But others, it's as normal as it is, that it should be metabolically stable, absorption should be good, solubility and flexibility is also important. So that is very vital that we look into it. Sorry about the background disturbance. My small boy is just, he's trying to find me out. Anyway, so moving forward, let us also look into P450. This is also one of our objectives that we wanted to look at the multi-parameter optimization. So when you look at this data, I have also already predicted, because it will take some time. So I already predicted just a minute. Yeah. So here, if you look at the P450 isoform of this compound, you will be able to see that it is trying to tell you which isoform. It is trying to tell you that which isoform of your P450, your compound is very selective or regioselective. So here you could see a major portion of a 3A4 is being bound. And it is also understood that we know that 3A4 is quite large site. So most of the compounds could go and bind there, being selective. But moving forward, when you look at the 3A4 isoform, you could see that the metabolic landscape is that this particular carbon is prone to break down like this. This nitrogen could break down like this. So it will be able to tell you what is the percentage of the metabolite it could form from this compound. Now, personally, if you ask me, I could say this particular nitrogen is also prone to break down to form metabolite also, or ionization can happen there too. So it is not that only predictions are going to help you. You should have a little bit more understanding on metabolic stability also. So this is something that we have to take care when you look at the P450 metabolic stability. So the stability values are predicted with composite side liability, where it is ranging from 0 to 1. Now 0 to 1 means any value which is lesser is more acceptable. Means if the composite side liability is lesser value, we tell that it is more stable. But if the composite side liability is more, that means closer to 1, that means it is unstable. So here what they are trying to tell here is, among the atoms in all these compounds, what the two atoms, basically the carbon 2 and the nitrogen 8, is very unstable. So being these two atoms are unstable, and that is why it is prone to form metabolites. And if you go below through these table, you will be able to see what are the possible metabolites on different atoms also. So for example, let me go through this. Yeah, C7. C7 is moderately unstable, but you can see there are a big chunk of metabolite that could form from this breakdown, and then it form a compound. So these kinds of analysis that you can do. Now, if you wanted to optimize the structure, the same thing can be done in NOVA. So you can go to NOVA and then mention the region, again transformations or match series, and then you can mention which region that you are looking at the breaking down. So I will just mention this particular region, sorry, this particular region, so that this will not change, only the other range side will change. And then I will say next. And then here I should have a better understanding, because basically here I'm not looking only at ADME properties, but I'm looking at a pro-drug strategy. So here I want to make sure that the metabolites are much more stable, it won't break down. So that is what I'm trying to see here with the applicability of transformation. And when I gave that, there is only few options available to me because of the chemical composition. So here if I say alkyl amine to amino alkyl carboxamide, that is what the transformation I can have. When coming here, I have methyl to tetrahydrofuran. So these are the kinds of transformation. Anyway, let's see what all the transformations possible, and then let us go for the property. So here I'm going to say that, let me find out the P450 table. Just a second. P4550. And this is going to take time. The only reason is because of, okay. Only reason is because it has to do quantum chemical calculation. So I don't know how many of you have read the paper from Professor Bharatam. Professor Bharatam also wrote a paper on similarly for quantum chemical calculations on this one, P450 metabolite prediction, right? So yeah, so anyway, I'm not going to do that calculation right now. So otherwise you can look at different property, diversity, as well as combination of property and diversities could be given. So you can say balance, how you want to balance between. So earlier in my slide, I was telling you I gave 25% to the score, and 75% to diversity. This is what you adjusted here. So you will be able to adjust the diversity as 75, yeah, and property as 25. So this will give you a balance on the prediction of new structures from the transformations. Or you can come up randomly, any new compounds come up, not selecting on specific structures. So first of all, you have to understand which region is affected, and based on that, we have to choose our design of molecules. So this is mainly the algorithm called as glowing molecule, which will be able to give you a lot more data about how this can be taken care further for designing of novel molecules. Additionally, we do have access to, so you can run Autodoc within StarDrop, which is something similar to Pyrex. So you have to generate your receptor in PDB-QT file, and you can have thousands or 5,000 molecules within StarDrop, and you can run Autodoc VINA so that it will be giving you the binding energy values. Serella is a module which is for deep learning and machine learning models, but yeah, I'm not going to show that because it has to connect to an online server. We do have AutoModeler, which is again for Qscr, and this is completely based upon machine learning methods, something like called the Gaussian process regression method. The reason why Gaussian process regression method is used is when I know everybody's, even my favorite is multiple linear regression method. But the problem with the linear regression method is that it has an issue of overfitting issues as well as false positives. So in order to avoid overfitting issue, always the best option is to consider partial least squares method or some machine learning methods. So that's why Gaussian process regression method and random forest methods are implemented here. By default, StarDrop will generate only 2D descriptors, as I already discussed. 3D descriptors you can bring in, and it is of your choice. You can bring in from Padel, you can bring in from RDKit, because that is your responsibility. Which 3D structure did you use for 3D descriptor calculation? Because of the bias, which I already mentioned to you earlier. So you need to know what you're taking for a 3D. Then bring the values here. You will be able to use machine learning methods to generate the models. So, but StarDrop itself will not generate 3D descriptors because of this bias, because we do not want to convert simply 2D to 3D structures and calculate the descriptors. And then, then there are other scripts where you can pull in e-molecules or enemy in databases. So you can search similar structures of your compounds and search the vendors and everyone from the e-molecules, ensure that you have account for e-molecules so that you will get the API token and then you can access it. And you can also, Divya Madam was asking earlier one question, if we have a compound, how to find similar binding hits or targets. So there is something called finding similar compounds, and you select a compound and click on protein data bank. So depending upon your internet speed, it might take time. So now I'm not doing it because I'm sharing my screen, it will take some time. So when you click on it, it will search all the RCSB co-crystal structures, and it will return back with the PDB IDs. So those PDB IDs, you can explore whether it could be a suitable binding hit targets. Doesn't mean that that is the disease target that you're looking at, but that is completely based upon a structure-based similarity of your compound with respect to the co-crystal structure which is there in the proteins. You can calculate ligand efficiency as well as other properties from Campbell. So you can search Campbell with the target IDs and other analysis. We have something called as a card view, where you will be able to do clustering and also neighborhood joining methods, which will be able to analyze the structure. So let me quickly show you one. Let me check just a second. Yeah. So let me quickly show you this for activity neighborhood. Property difference, yes. Yeah, this is what I was telling you initially during my presentation. The software will be able to analyze from this particular compound to this compound. The only difference here is you can see at both the sides, the positions of, so here you have another keto group with methoxy. No, two methyl groups are there. Yeah. So here you don't have the difference in the activity. It is 0.534. And again, the difference in ADME properties are also given. Similarly, you can also look at each functional group if they are exactly similar, because I don't have a similar structure here. That's why it is not able to give, because I have a very diverse structure here. So it will create also a spiral network, which tries to tell you that if this compound is a reference molecule I have chosen, which is a closest other compound which is having similar structure as well as closest activity data. So both can be predicted. And similarly, if I choose another compound as my reference molecule and I'm asking matched pair neighborhood, and then I'll say this is my activity data, or I will take this estimated data, and then I click OK. So it will be able to now generate. As I said, I have a very diverse set, so that's why it couldn't tell you, but it quickly told me the difference between this particular derivative and this derivative in the biological activity data is 1.66. And when you're coming to solubility data, it is 0.181. So this is the structure. This is the complete structure at the below you can see. So if it is a homogeneous database you are going to load, of course, you will be able to see many derivatives like this in the table to tell you what is the difference between the activity as well as in the solubility. This is what you have to write in your discussion when you say, when you predict ADME, that because of this particular functional group in this compound, this exhibited a better solubility when compared to other compounds with other function group. And this helps the reader for him to implement new ideas for their project. See, publications are transition of ideas, and each level it has to go up. So just saying the solubility value, what is the use for them? But when you tell about very narrow down to functional groups or derivatives and tell them this is how it has increased or this is how it has decreased, they can immediately implement it on their work also, maybe without a software also. So that is what we have to look at, the research transformation of the data that we are going to transmit. Right, so this is all about StarDrop, which I wanted to mention. But before moving forward to the other software, which we will be discussing about docking, I want to know from the organizers how much time we have more, and I can address a few more questions.
Yes, sir. Sir, Giri sir, you can have another 10-15 minutes time.
Great. Thank you so much, sir. So before that, I will complete this and I'll take the questions if any. So quickly, I'll go with the Qscr because Qscr is if you look as a person outside, like every docking program, you will feel it is a docking program. But for me, it is more than that. I'll try to explain it and see how we can convince it. So this is biotin. I have taken one STP, which is a heat shock protein, and defining this compound as my reference or co-crystal. Automatically software identified the closest amino acids, and at the bottom you can see it is being highlighted here. Okay, now let's imagine I don't want this. I will do it myself, because most of the time I see people complain that when I use tutorials it works, but when I use my protein it doesn't work. So let me try to do with the same thing without defining my co-crystal. So I will say do not extract the ligand from my co-crystal. Okay, I want to consider this as an hypothetical protein. How do I treat it? So I'm saying do not extract a ligand, and I define, okay, I have only protein, I don't have ligand. Okay, I removed it manually. And then before moving forward, I told about conservation studies. So I will add this to my protein editor mode, and then I'll click this button, which is called, it will search online to use a server. But before that, we need to know the binding site. So what it tries to do is that it will search only the binding site of your protein with other binding site of the other proteins in the whole RCSB database. So to do that, let me go back to proteins. These are the different modes available in Qscr. proteins, protein binding site, binding editor. You can mutate it. You can make flipping of amino acids. All those things can be done in protein editor. Then you have analyzer to analyze your trend between the binding affinity calculated by the software and the binding affinity that you achieved through wet lab studies. It is not a correlation study. It is a trend analysis. For a chemist, you can do molecular editor, and you can do fragment-based analysis using Inspirator, and you can do covalent docking, you can do normal docking, and you can do template docking. So covalent docking is something that is recently being implemented. So when you're doing covalent docking, make sure that you know the R group, and you have to define the R group which has to form the covalent bond. Now, I'll add this particular ligand to the binding site. Now, this is where the question is. When you don't have a co-crystal ligand, the Qscr will use an algorithm called as dock site, and it will be able to predict all the possible pockets available in there. By the way, I have to remove the ligand. So let me do that, just a second. So I have to, in order to remove the ligand, I can come here and I can remove this biotin. So I will go in editor mode, click the right button and say delete component, and I will add it here. So this STP2 does not have ligand in it. So I will add this back to the protein mode. I will delete this. This I'm doing it the other way to just show you how do you handle a protein without a ligand. So this is my protein without a ligand. Okay. So now I'm going to binding site. Now automatically it predicted that this is my pocket, because when the ligand was there, it said there is no pocket. So it identified the pocket. There might be more than 1 or 2 pockets, but in this protein, there was only 1 pocket. Now I click on this and say add surrounding residues. So to be able to find out the pocket. At the bottom, if you click on this residues and scroll, you will be able to see at the bottom, you can see there are some amino acids with highlighted in cyan or magenta, whatever color you call it. So, or pink. Now, if you think that you want this amino acid, so when I highlight, you can see on the 3D window, it is highlighting. If you think that this amino acid also to be included into your binding pocket, click the right button on top of it and say add residue to binding site. You can include that too. But tomorrow you think based upon your literature data and another analysis, you understood that this compound, this amino acid not required, you can also remove from the pocket. So pocket modifications can be done. But when you modify, make sure that you have a solid proven evidence that why you wanted to include that amino acid. Once you are done, you have to click this play button that makes sure that the binding site is defined, and automatically software identifies all the water molecules. Water molecules can be considered for the docking here. It's your choice. Software has no limitations like others. So you can choose you can select or unselect water molecules for your docking. Now I have to go to my docking program, and then I can see my pocket with lipophilicity. And this is my pocket here, right? Now I can do the docking. I can load molecules from, maybe I have some molecules already. So I can load them and I can do docking. When I'm doing docking, make sure that you choose, by default, the pose number of poses generated will be 10. But if your molecules are too flexible, too much rotatable bonds are there, make sure you at least give 40 as poses. Simply giving 500 and all doesn't make any sense. So make sure that at least you give 44 rotatable bonds. Here, interest of time and speed, I'm reducing the pose. One more biggest advantage with Qscr is that, let's say you have a machine with 16 core or 64 or 32 core CPU machines, and you wanted to do a virtual screening against binding DB. The binding DB single SDF file itself will be 6 GB. Okay, so keep that in mind. So 6 GB means it's huge. So you can do that on a multiple CPU. So Qscr has a capability to run your virtual screening on a parallel mode on all the 64 or 32 or 16 core. Now, if you're running a laptop with 4 core, it will run on all 4 core. So that's what is the requirement is. Otherwise, there is no heavy requirements, but completely depending upon the file or the file size of the ligand that you're going to choose. Then I click on docking. So this is the normal docking which I'm going to do. So what the software is now trying to do is it is trying to pre-process it and then trying to generate the normal docking poses. Then it will try to calculate hydrogen bond interactions. Then it will calculate binding affinity based upon Hyde algorithm. It will try to understand the clashes if there is any intra or interligand clashes. And then finally, it will calculate the torsions. Torsions means from the dihedrals, how far the conformations that is being generated by the software can form a co-crystal and which cannot form a co-crystal. So this information, how it is going to helpful for a chemist, because it will tell you where or which region of the molecule you have to optimize or make modifications in order to enrich the activity or the affinity data. Now, let's imagine that the calculation has started and you wanted to shut down the computer because power has gone. So whatever now 12%, 13% is coming, if I cancel it, whatever the docking has been done, it will automatically load that data. So I'm just now canceled it. So now whatever docking has been completed, it will load it. So it is not that it will, I think I didn't cancel it. Let me cancel once again. Yeah. So whatever it has completed, all the data will be loaded to your results page. So now it is only generating the docking. It did not do any binding affinity calculations and all. So wherever you are canceling, those data will be retained in your data. So that is possible. And other thing is that you can, I think it didn't cancel. I think, oh, okay, now only I canceled it. Now only I canceled it, sorry. So yeah, so likewise it can be done. And then it will also show you unoccupied exploratory spaces. Means when you, after the docking, it will be able to tell you which all the regions of your structure or a chemical structure that you can further explore to make derivatives. Where are the spaces available within the conformation of the pocket? And also you can define pharmacophore constraints also. Let's say, as I told you, you know that you need to have an interaction with, let's say, aspartic acid 29. So without that interaction, I cannot accept any docking results here. So you can define a receptor-based pharmacophore constraints mentioning that there should be an interaction at aspartic acid. So when I canceled it, whatever, so only 29% the job ran. So whatever it has done, it has been reported here. These are some of the natural compounds. So see, you can see it is interacting. Now, if you think that you wanted to do the binding affinity now for selective compounds, you can select them and then go to this calculator and say estimated affinity. So it will now calculate the binding affinity calculations also. Right. Now, then I am ranking them based upon the affinity. So here, estimated affinity is between nanomolar and micromolar. If you're very adamant that I want to see Qscr value, I want to see estimated affinity value, of course, we have a provision to generate the Qscr as well as the affinity value in micro or nanomolar. But to be honest with you, do you think that is going to help you? No, because these values can never replicate to your in vitro studies. Always the range is going to help you a lot, whether my compound is within the range of nanomolar and micromolar. And these are very well accepted in JMedChem Jack's paper. I have more than 10-40 papers, 24 papers already from different authors. They have already used this methodology. We have to change our mindset. We have to come up with at least a perspective on how we look at these docking results. Now, the interesting part here is that when you click on these green circles, you will be able to see some energy contribution of that particular atom. And that is very important. So here it is saying this, this atom is contributing minus 1.6 kilojoules, and this carbon is contributing minus 2.6 kilojoules. I can switch off my binding site. I can see the interactions where it is coming, and then I can label it also. So I label this one is, I can change the size of the label. I can take a screenshot. All those things are possible. Even I can take a 2D depiction using a post view tool. So that is also possible. And now if I wanted to do torsion analysis in order to understand how far these are bioactive conformations, so I will enable torsion. So you can see molecular clashes showing between the ligand and the receptor. In some it is there. The clashes are not always good. Now, what I'm going to do is I'll enable the site and then I click on this torsion dihydral. So here it is showing that this is in green color, means as per Cambridge Structural Database, this is in the acceptable range. This particular torsion is in the acceptable range. But when I come here, there's an orange color. This particular bond or the dihedral or the torsion is an unusual area of crystallography or the crystal structures which is present in Cambridge Structural Database. These are the set of informations that you get. But earlier I told you something, unoccupied spaces. So you can see now there are some cloud, black or gray colored cloud, which helps to a chemist to understand, okay, these are the regions I can think of making more derivatives. There are spaces available. You shouldn't simply make derivatives here and there. By seeing a docked result, that's where you have to think of where you can make derivatives and design new molecules. Now let's imagine I want to make some modifications here. So I will take this compound and I will go to molecular editor now. Now this is a molecular editor window. If I want to replace anything, so I saw that I can add some molecule or atoms this side. So let me make a derivative here. So I will select the hydrogen atom here, okay? And then I'm going to replace this with any other ring I can do, or I can change the element. So what I will do is I'll change the element to maybe chlorine. So it has changed to chlorine now. But automatically software is saying, okay, there might be clashes if you make it as a chlorine. Do you want to reset it? Yeah, I'll reset it. I'll again select, and then I will say, I will think about oxygen. Yeah, that is kind of okay. We have to redock and see if it is fine or not. And then we wanted to see that this particular bond is not that acceptable. Can we try to turn around and see? So I can click this to rotate and flip. So I can rotate. You can see that it is rotating. See, you can rotate and see whether there is a change in the torsions happening or not. Here you can see now it has become green, but it is showing an intra-ligand clashes. But again, we have to dock and see. So let us quickly dock and see. So here I'm going to do a template docking quickly to see whether the addition of hydroxyl group and this is going to improve or not. Let us see that. Let's a binding affinity calculation also finish, and then we can see. So this is the kind of optimization that we have to see when you're designing new molecules based upon docking data. Okay, some docking results have come. Let's see. The affinity has not improved when compared to the original molecule. The reason is because we have clashes here. It's just because of the clashes. But otherwise, the OH group is acceptable, no problem, right? But the other one has clashes. And this one, there is no clash, but still the affinity did not improve, right? There are some interligand clashes also. So when you design new molecules or design new derivatives, you will have a more vicinity of understanding how this is going to affect the stability and affinity of the compound. So this way also Qscr can be looked at. Now, let's look another perspective. You can go to a fragment based drug design. By this, I will finish off. So I wanted to see whether I can replace this particular region. So I will try to replace probably this region. Yeah, I clicked it. So this particular region, I want to replace it, right? So then here you can see the option automatically came called as growing. So if you have a small ligand in a big pocket and you want to expand it or grow it, you can look at it. When I click on it, it will search. I'm using PDB fragment database. So it will do a fragment growing, and it can also perform docking and finally give you the results. So based upon here, I have not given any pharmacophore constraints, but I can define pharmacophore constraints saying that in this particular, close to this amino acid, I should have an acceptor. In another amino acid, I should have a like donor. In another place, I'm saying I should have only either nitrogen or oxygen. So I can define the constraints, and it will not go out of the pocket also, because the pocket itself will be a constraint for the shape. So these kinds of parameters can be given for fragment-based drug designing, and it will be able to come up with more fragments and composites. Now, defining which interactions it should have, you should know earlier that what are the key amino acids it should interact, which atom it should interact, what kind of pharmacophore properties that particular compound should come up. Based on that constraints, it will be searching all possible fragments. Now, it came up with plenty of fragments. Let us see. So you can see it replaced with some few other different molecules. Of course, we have to do docking, but it just replaced it based upon the shape of the pocket, right? So then we have to take all this to our docking panel, and then we have to perform the docking again. It will take time, so I'm not repeating that. But in order to define the pharmacophore, this is what you have to do. So here the pharmacophore, define the constraint. Let me switch off the binding pocket, and then I can say, for example, I'm saying I need an interaction in this particular amino acid, okay? And then I'm telling I need any nitrogen or oxygen, or I need an aromatic ring very close to it, or I need an hydrophobic interaction. Without that, I don't want any docking result. That way you can define if you're very confident on what you're trying to do. So for large scale virtual screening, this is really helpful because we do not want to unnecessarily go and sit and analyze after the score, whether it is interacting with this amino acid or not. Rather, we give this condition constraints and then later, because we have to be very sure based upon literature data and other analysis that our compound should have an interaction with this amino. Otherwise, I cannot accept this mechanism. So like this, different perspectives are there. But when you check online for the tutorial, these are all not there. So this is all depends upon the project, you will be able to implement that. So based on this, that's all from me for today. But if you have any other questions or any comments, I'm happy to take it. Thank you so much. Thank you so much for your patience and also responding to many of my questions and very wonderful questions from your side too. So, and I also would like to thank for this great opportunity. Thank you.
Thank you, sir. Giri, sir. It was very helpful for our students and research scholars and staff members. So actually, you talked about what are the key aspects of this drug design, especially taking multi-parameter into the approach. So in the lead optimization of the drug candidates. So very nicely illustrated with your all the presentations and with your skills, everything was amazing, and it was very useful for us to learn like that. So if there are any questions, quickly we can take up and further we can wind up. Dear participants, you have to give the feedback in the Google Forms that we are given, and after that you'll be receiving the certificates to your registered email IDs like that. Okay, fine. Thank you very much. If there are any questions.
Okay. So a few questions are there. Let me read them. Is there any free tool to find similar co-crystal ligand with our structure? With your structure means proteins or, yeah, that's what I told you, HitPick server. HitPick server is an online tool for your chemical structure. You can submit the SMILES. You will be able to find the similar co-crystal PDB IDs. From the PDB ID, you will be able to find the co-crystal ligands. Yes, it is possible. If a molecule after docking binding affinity more than -10, dock score or more than -10, the particular model is having toxicity. Okay, you mean to say that even if your affinity is better, but still you have toxicity? Yes, you have to optimize it. If you are a chemist, you have to do it. Protonation in Qscr is being done by using PROTOS. So it takes care of addition of hydrogens, including the orientation of the hydrogen, so that it is in par with the X-ray crystal structure. So based on that, Qscr uses the protonation. And for the ligand molecule, if you are loading, it can load as a SMILES or a 2D structure. And then it will be using in-house 3D ring conformer algorithm in order to generate the 3D structure, optimize them, as well as generate the ring conformations. Because when you have a cyclohexane and all, we have to see whether it is a boat or a chair conformations. So ring conformations will be also taken care within the Qscr. Any other questions? Oh yeah. Can molecular dynamics be performed in the cloud? Yes. Let me show you quickly. Just a second. What you have to do is you have to go to this GitHub link, which I have already given you earlier, and go to this MD notebooks. Under MD notebooks, go to Colab. And under Colab, you will find an icon here, open in Colab. Click on this. You can do molecular dynamics using GROMACS in Google Colab on the cloud. But every 12 hours, it is going to reset. This will work on your Chrome browser in mobile also. So when you take this, take a copy of it and work. This is on my Google account, but when you open it, it will be on your Google account. All the GROMACS commands, everything, compilation, everything I have already set. And this particular notebook was 2 weeks back cited by GROMACS developer itself. So they cited saying that this will be very useful for learning purpose. So they validated it. So you can use it with guarantee. So all the commands are here for solvation, production run. All the files are taken from my GitHub for the parameter files and result analysis. Everything is given here. So for learning purpose, I would really recommend this. But for production run, I told you, every 12 hours, it is going to reset. So that will be a challenge, even though the performance per day, it is 154 nanoseconds per day. Then how molecular dynamics using can be for, sorry, I don't know. Desmond on cloud, I don't think so. Because they are giving it for free. You have to try it on your computer. Desmond has its own challenges to run it on cloud, especially on the Google Colab. So I really don't know. Can SID also be generated? Can you tell me what is this SID? Sorry, are you referring to the PubChem's ID or I don't know. Can you please tell me what is this SID? Simulation interaction diagram. Yes, you can generate using PostView. That's what I said. Using PostView you can do. But for the molecular dynamics especially, you can use the Colab, where I have integrated Matplotlib and MD Triage and MD analysis to generate the interaction diagrams. Yes. Binding affinity score must be below -10, or based on the other parameters we can conclude. Kavita, please do not confirm that you should have binding affinity less than -7, -10, nothing like that. As I told you earlier, relatively among your data set, if you select a 10 compounds, 10,000 compounds, so 1 lakh compounds, among that, which is the best? You pick up that. Now, if you think that, okay, I selected all, but my binding affinity is -30 kcal per mole, then take -13. If some came -2, take -2. If you think that -2 is not acceptable, because based upon our understanding and experience, then go for expanding your number of compounds more and then further do analysis to see whether you can have an increase in the enrichment on the energy value. But if you say -10, again, the data set, whatever you selected doesn't come within that, you have to again consider different set of compounds, right? So make sure that your initial set for screening is a bit larger and the space is much more broader so that you can end up with all your threshold criteria. Now, that is my take on that. So I don't see any other questions per se. Oh yeah. Do you recommend performing IFT for a wide variety of natural products post docking before MD? Of course, yes. If you do have a good amount of hardware and time, I absolutely recommend you to do that.
Okay, with this, I think no more questions. Now I can think that it is time is already 1:04, like that. So better, I would like to conclude this seminar with the formal word of thanks from Dr. Yogesh Kumar, our colleague.
Okay. Thank you, sir. Thank you, Prashant, sir. Very good afternoon, everyone. It's my privilege to propose vote of thanks in today's virtual seminar on the title, Lead Optimization of Drug Candidates Through Multiple Parameter Approach. Firstly, I profoundly thank our today's resource person, Dr. Girinath G. Pillai, Jastra Innovations Bangalore, for sharing his vast experience, mainly considering multi-parameter approach, that is not only the docking interactions or any other that is amino acid residues is important. Rather, you have to consider the potency, viability, stability, and quality with diversification, etc. And also, sir covered about the importance of ML in drug discovery rather than that is artificial intelligence. Thank you very much, sir, Girinath sir, for sharing your experience. And I sincerely thank Dr. B. Suresh sir, Pro Chancellor, and Dr. Surinder Singh, Vice Chancellor, JSSCHR, for their support in organizing today's virtual seminar. And I extend my thanks to our principal sir, Dr. T. M. Pramod Kumar, for his encouragement for successfully organizing this seminar. I would like to thank our head of the department, Dr. G. V. Pujar sir, to support in successfully organizing this virtual seminar. And yes, I would like to thank the organizing secretary, that is Dr. B.R. Prashant Kumar sir, for his initiative and meticulously planning to organize the virtual seminar. And also, I would like to thank our faculty members for their kind support and cooperation. And I would like to thank Dr. Ravindra sir, Chief Information Officer, JSSHER, for providing the platform and coordination throughout the seminar for smooth conducting the seminar. Finally, we thank our all enthusiastic participants for patience listening, without whom I think this seminar would be incomplete. Thank you, all participants, and once again, thank you all. Thank you, one and all.
Yes, thank you, Yogesh Kumar, for the vote of thanks. And as the organizing secretary for this virtual seminar on lead optimization of drug candidates using multi-parameter approach, so I wholeheartedly thank all the participants who are gathered on this platform and learned how it can be done. And we kindly request you to inculcate this knowledge in your research and do better research and publish it in good journals like that. So all the hints were being given by Dr. Girinath Pillai. So I thank Dr. Girinath Pillai as well as the participants who have gathered on these platforms. Okay, so thank you very much. So with this, we conclude where we come to the end of this seminar. I declare that it is the end of it. Okay, thank you very much.
Thank you so much, sir. Stay safe. Bye.
Thank you, sir. Thank you. Thank you very much.